is that? That's because, right, like it kind of alludes to in the passage, these bonds are high energy because of the associated electronegative charges exerting a repelling force between the phosphate groups. Okay, so you can imagine on your ATP, right? It's when you have these two phosphate groups next to each other, it's very unstable and would love to get rid of that. Okay, so it turns out that if you're going from ATP to ADP, right, it's getting rid of phosphate group, so it's going to split off a phosphate. If you're going from ADP to ANP, on ADP, you still have two phosphate groups, right? And so those two phosphate groups still don't want to be next to each other. And so that's why the delta G is very similar. And then adenosine triphosphate to AMP plus PP, again, it's very similar, right? Okay. And similarly, to just even go further, right, if you have PP to 2PI, right, what's the delta G? Does anybody want to take a guess on that? It's going to also be very similar. It's going to be something like 32. Okay. What about if I were to do this one? What about AMP to adenosine plus PI? What do you think that delta G would be? Bigger or smaller than all these? Right? It would be, well, technically bigger. It would be less negative, right? Less negative. So it would actually be, I think it's around negative 9.2. Okay, and that's because this phosphate is by itself, so it's actually a little bit more stable. Okay. And again, you can kind of see this over here in this little diagram, right, where you can collect energy from the PP breaking up, right? You can collect energy going down to AMP, and you can collect, energy going down, or the release of energy, sorry, going into ADP. Okay. Okay, so on your MCAT, and they might try to give you one of these tricky questions, right, where they're comparing things, and you obviously all have this assumption that ATP to ADP, right, or this one right here, that ATP to ANP would be bigger. Now, you guys had a really good guess. You guys would be correct, and I want to emphasize this. If it was ATP, I'm going to write it down here, to 2PI. You know why? Because that's coupling ATP, that's two reactions, ATP to PP, sorry. And then that's also coupling PP to 2PI. So you actually get double the energy. You would get that, negative 33 and then that negative 32. So it'd be two reactions, and you can, have two energies. What is P versus PI? So PP, right? That's this right here, pyrophosphate. And PI equals the phosphate group, which is just O, right? So it'd be one of these groups. Does that make sense? Yes, and so the logic would be exactly that. B, C, and D are all similar, which would rule them out. But the actual real logic is you need to know glycolysis. And this is why this is an unfair question, because we haven't covered it. But phosphoenolpyruvate to pyruvate actually generates an ATP, right? So for this reaction to be favored, right, it's better have a larger negative delta G, so you can actually generate an ATP out of it, okay? Are you guys seeing that? Okay, and question 2, right? This one's just pretty easy again if we know glycolysis, all right? And we'll know that PFK, right, burns an ATP. Right, converting F6P into F16P. And we're going to go over glycolysis in big details later, okay? Yes, Melody, exactly. No, it says, so more energy is retained in D because both phosphates remain together with A and P. Yes, exactly. So this is a very technical thing, right? If you look right here in D, these things are still connected, right? That's still a pyrophosphate. Now, if you were to split pyrophosphate into 2pi, you're going to gain even more energy, right? Which is shown actually down here, right? Into 2pi. Do you see that? Great question. Great question. Okay. Can we move on?
So just to confirm, D releases less energy than B and C, or more?
Are you B?
D, sorry, delta. D for delta.
Oh, this one? It actually releases slightly more, but it's very similar. You know, negative 33 versus negative 35. And it's so close that you should consider them pretty much the same, right? You wouldn't get a question that is like that, right?
I'm confused though. Isn't more negative number more energy released?
Yes, the bigger the negative, the more energy is released, right? So if we go back to this, right, the bigger this negative number is, the more energy is being released, right? Like here.
Yeah, but isn't that less energy than B and C?
For which one?
B and C are negative 35, so is that a higher negative number than negative 33, which is B?
Yes, so it would release slightly less, right? So it's negative, it's a smaller negative number, right? Which is a slightly bigger number, which means it releases less energy.
Yeah, isn't the question releases most energy or more energy if you take out A?
Hold on, your mic is pretty garbled.
Doesn't D, if the question is releases most energy, and we're not considering A, then isn't D a less negative energy, less negative number, negative
So it would
33?
release less energy. The higher the negative delta G, the more energy is released, right? Negative 6,000 is release of more energy than negative 600. More energy released.
So then here, B and C release more energy than D.
Yes, release more energy than D technically, but they're so close to each other. They're so close.
Okay.
33 versus negative 35, right?
Yeah.
This one is negative 62. This releases way more energy. That's why A is the right answer.
Got it. That was my question. Okay. I was just confused why we were circling D when it was less energy.
Sorry.
Never mind.
Yeah.
No,
It's
you're
all the scribbling. It's because I was trying to prove a point here, right? That if we were to eliminate this one, it'd be really hard to come up with the right answer because they're all so similar, right?
Yep. Yeah, that makes sense. Yep. I'm with you.
Cool.
Josh,
Okay.
I have a question for number 2.
Yes.
The passage clearly states that ATP inhibits PFK1.
Now you're getting ahead of yourself yet again, right? ATP, this we're going to get into this when we talk about glycolysis, right? But glycolysis is very regulated by the amount of energy, aka ATP, floating around in the cell, right? So it needs ATP to convert, right, fructose 6 to fructose 1,6, right? You're adding a phosphate group onto fructose, and we're going to get into this, right? But if your body has a lot of ATP, right, then it doesn't need to make more ATP, and so ATP actually acts as an allosteric inhibitor of fructose 6 of PFK, Okay, and we're going to draw this all out, the mechanisms, the equation, so that'll become good. But you're thinking well. That's good. Okay.
So is question 2 asking which activates or inhibits PFK1?
No, it's asking about the coenzyme. Okay, so a coenzyme like ATP, right, is just carrying a phosphate group. So you can imagine, let's go up here. You can imagine, right, fructose 1,6-phosphate, right, is like this, and it's got a whatever CHO up here, you know, OHs, H, O, right? And if it's fructose 6-phosphate, you have a phosphate group down here, right? You can imagine this. And if you're going into fructose 1,6-phosphate, right, where you have a phosphate group here, oops, you get a phosphate group added here, right? And here's your double bond O still, right? I'm just drawing this very rudimentary, right? And you have a phosphate group down here at the bottom, right? Actually, this phosphate group is right here. You erase that, right? You're adding a phosphate group. Are you with me? You have to use an ATP to add a phosphate and you get an ADP. Does that make sense? And this is PFK. Does that make sense? So you're adding a phosphate group, right? You're adding another one that's on this ATP up to the carbon 1 of fructose 6-phosphate to make it fructose 1,6-phosphate. Okay.
Okay, so quick question. If
Yeah.
we are looking at the passage alone, say you're in the middle of the MCAT, you know, have a blank in the moment about the specifics of glycolysis. Using the passage alone, how would we find that answer?
The answer to question 2, I'm not going to
Yes.
get it. You're going to need to know that. And you're absolutely going to need to know that for the MCAT. Right. And I mean, glycolysis, especially out of all the metabolic pathways, you need to know inside and out. Right. And we're going to get into that when we start talking about glycolysis, but you would need to know that. Right. And so this passage right now feels very unfair, right? Because we haven't talked about glycolysis, but we're starting to work into it. We're starting to get you to think why all these things are relevant. We're talking about energy here, right? Cool. Okay, this is just a quick slide. We kind of already talked about this, right? But if you have like an acetic or a phosphoric anhydride, right, you get these negative partial negatives, right? And they're going to repel each other. So they're going to actually want to split apart, right? So you could also just imagine this as, you know, your phosphate groups, right? If it's a phosphoric anhydride, right? These negative charges are going to repel each other. And so you actually will want to split these up. Okay, and that's why there's a lot of energy released when this happens. Okay, similarly, another thing is resonance when there's competing resonance structures. So if you can imagine that not only does this resonance structure is possible, but this one is at the exact same time, and that'll create a strain, meaning that, you know, it'll favor the products if you were to, you know, split this up. Okay. And yes, the correct answer to question 2 is A. Okay. Okay. Let's transition now into Michaelis-Menten kinetics. Okay. And so we're going to go back to the very first slide of the lecture, right? So now that we know what delta G is, we can start talking about, you know, enzyme catalysis. Okay. So again, enzymes do not alter the equilibria, right? They can accelerate the forward and reverse reactions, and they do this by stabilizing the transition state, right? So it can actually lower the activation energy. Okay. And so with enzymes, we care about the rate of the reaction. We care about the kinetics. All right. So here's a you know, the simple enzyme plus your substrate, right, goes to this enzyme substrate complex, which then can go back to your enzyme and then, release a product. Okay. And so when we're talking about enzymes, catalyzing reactions, we care about how fast they're able to make products. Okay. In other words, we care about, the velocity. Okay. We care about the velocity of the reaction. Okay. And in a biological system, at least for our model, we're going to assume that the enzyme concentration is fixed. Right? There's just a fixed amount of enzyme in your body, and this will allow us to make a model. Okay. And what that model is, right, if we just, take this, and, write it down, enzyme plus substrate, goes to the enzyme substrate complex, which then can turn into your enzyme plus your product. Okay, here's our simple substrate to product model. And so what I'm going to do is I'm going to label each of these arrows as different rates, and I'm going to call this one K1. I'm going to call this one K2, and I'm going to call this one K3. Okay. Okay, Tiffany, you're getting way ahead. We're going to cover that coming up here soon. Okay. So we're just slowing down here, talking about, we're defining our substrate to product model, and we're labeling each of these arrows, right, as these rates K1, K2, and K3. Okay. And so, again, for a biological system, we're going to assume that our enzyme concentration is fixed. And because of that, then for many enzymes, right, the rate of catalysis V is going to be able to vary based on the substrate concentration. Okay, and you'll get this nice parabola. Okay, and so we care about velocity, right? But what exactly is velocity? Okay, velocity is how much product we can make. So it's from the enzyme substrate complex, right? How quickly we can make product. So in other words, velocity V is going to equal our K3 rate times the concentration of enzyme substrate complex. All right. And so we're going to get back to this, but if you notice on this graph, right, if you have a very low substrate concentration, right, you can see that this is almost linear. Okay, so, in a fixed T, when the substrate concentration is small, right, you can see that the velocity is almost linearly proportional to the substrate concentration, right? And this is, going to be what we call first order kinetics. And conversely, if you start to increase your substrate concentration, right, when the substrate concentration is high, you'll notice that the velocity is nearly independent of the substrate concentration, right? And so this is going to follow zero order kinetics. Okay. And so, we can visualize, V graphically, right? And we're going to be revisiting this graph in just a second. Okay. But we want to look at what all of these parameters mean. Excuse me. We want to look at what velocity really means, what the Vmax really means, what Vmax over 2 really means, what Km really means, what substrate concentration really means. Okay. So now we're going to go through that. All right. So here's our little simple model. And what model are we interested in? We are interested in something called a steady state model. All right. And this is where the concentrations. Yes, no, you are not crazy. We're going to get into that. That's called a Lineweaver-Burke plot. Just hang in there. Okay. We're trying to describe why all these parameters make sense. So when you look at a Lineweaver-Burke plot, you'll be able to make sense of it intuitively, right? Not just memorizing what each thing does. Okay, we're trying to understand where these come from. Okay, and so when we're looking at this, we're interested in something called a steady state model. Okay, and that's where the concentrations of the intermediates stay the same. So when I say intermediates, I mean the enzyme substrate complex, right? And where the reactions of your substrate and your products are constantly changing. Okay, so in other words, what we really care about, right, is the rate of formation of ES, right? And then the rate of breakdown of ES. Okay. So I'll write that here. Yeah, rate of formation of ES. And you can look at this, right? So how do you form ES? Right? It's K1, this rate this way, times enzyme and substrate, right? Which can be represented here. And what about the rate of breakdown of the enzyme substrate complex, right? Well, that's just your rate that's going back to your enzyme plus your substrate, and then your rate that's going from enzyme into your actual product. And you can express that down here, okay? And so at steady state, what that means is that we can actually set this equation equal to this equation, right? And you can rewrite that, right, as K1 of, oops. Of ES, of enzyme concentration times substrate concentration equals, right, K2 plus K3, right, your rate of breakdown times your enzyme substrate concentration. Okay, you're just setting these equal to each other. Okay. And so if you do that, right, then you'll see that you have your rate of formation, right, K1 going to here, and then your rate of breakdown, K2 and K3. All right. And so now if we wanted to solve for the concentration of your enzyme substrate complex, you can rearrange the equation and get enzyme times substrate all over K2 plus K3 divided by K1, right? And this is now where we're going to introduce something called Km, which is the Michaelis-Menten constant. And what that is you take this and you just turn it into this. And let's pause here. What is this really saying? Okay. If Km is equal to K2 plus K3 over K1, right, a good way of thinking about this is essentially saying, right, the constant or whatever rate of breakdown of ES over formation. Okay. And this is called the Michaelis-Menten constant, Km. All right. And now that we have, you know, this for ES, for your enzyme substrate concentration, an equation for it, you can actually, and we're not going to do this in this class, it's very long and confusing and, you know, past the scope. You can use this to get to your Michaelis-Menten equation. And if you are interested, you can see it like actually in the bio books, right? Which relates this back to your enzyme velocity, right? Your actual rate, V. And you can write that as V equals Vmax, right? Times your substrate concentration over your Michaelis constant plus substrate concentration. All right. And again, kind of like we did before, the best way to, you know, understand an equation like this is to just start plugging in the extremes, right? And, you know, noticing what happens to, you know, the reaction velocity under different extreme parameters. So what happens, right? If our Km is much, much, much greater than our substrate concentration. Okay. That essentially eliminates this parameter, right? And so you're left with velocity equal to Vmax times S over Km. And thus your rate is proportional to your substrate concentration, right? So look at this, right? What that is saying is kind of proving your first order kinetics. All right, so that's where that came from. Similarly, what happens when your substrate concentration is way bigger than your Km? What does that do? That essentially eliminates Km, right? Which means just Vmax times, substrate over substrate, Vmax over 1, right? So your velocity will just equal your Vmax, right? Which is saying rate is independent of your substrate concentration. And so if you go back over here, that's going to correspond to your zero order kinetics. All right. And so what happens if we test right in the middle, right? What happens when our substrate concentration is actually equal to our Michaelis-Menten constant? All right. That would just be V equals Vmax over 1 over 1 plus 1 is 2, right? So your velocity is going to equal to Vmax over 2. All right. So in other words, excuse me, right? Km is equal to the substrate concentration at which the reaction rate is half the maximum value. Okay. And does somebody have any questions here? Okay. What part are you guys unfamiliar with? The first two on this page or the last page? You're talking about testing the extremes. So this is the Michaelis-Menten equation. Okay, so do you see this equation? This equation derived from this steady state model that we just went through. This is the equation that gives us this graph right here. If you plug things into that equation, they get plotted on this graph in this manner. All right. And so if you test the extremes of this equation, right, either when the substrate concentration is way bigger than the Km, or vice versa, the Km is way bigger than the substrate concentration, you're testing the extremes of this, right? So you're testing it in this region, and then you're going to test it in this region. And here you'll see that they're proportional to each other. And here you'll see that they're independent of each other, right? Over here, if you keep increasing the substrate concentration, it's not going to do much to your velocity. Versus over here, it's very proportional. You increase the substrate concentration, you increase the velocity. Does that help you? Yeah, this stuff is very technical. And you're going to get a lot of information right now, and you're going to just need to practice, right? And go back through these slides or whatever you need to do, okay? And what we're trying to do is we're trying to break all these parameters down. What most people do on the MCAT is they try to memorize these parameters, and sure, that's good, and maybe it'll work, but you're going to get weird situations, and having an understanding of where they came from and what they actually mean, when you get presented a question, it'll really help you, understand it better. Okay. And so where were we? Okay, here. And so again, we just plugged in our substrate concentration and we just set it equal to Km, right? And that shows that V is equal to Vmax over 2, right? So when the Km is equal to substrate concentration, right, that's going to be where the reaction velocity is at half of Vmax. So back up to here, that's going to be this, right? So Km is equal to velocity is equal to the substrate concentration where velocity is half of Vmax. Okay? That's where that comes from. All right. Okay, so now let's suppose again that K2 over here is much, much, much bigger than K3. In other words, that means like when the enzyme and substrate form a complex, instead of going to a product, it just dissociates really quickly, right? You form enzyme substrate, it doesn't make the product, it just goes back to enzyme plus substrate. Okay? Right? So thus, right, your enzyme substrate complex dissociation is much more rapid than your enzyme plus product formation. We're going this direction, right? So in other words, when K2 is much, much, much greater than your K3, right? Your Km is going to equal the dissociation of your enzyme substrate complex back into enzyme plus substrate. Okay, and this is important. Therefore, what Km really tells us, it's a measure of the strength of the enzyme substrate complex, how well those things bind to each other. All right. And so I'm going to double box this. If you have a high Km, right, that's going to give you a weak binding of the enzyme substrate complex. And if you have a low Km, that's going to give you a strong binding of the enzyme substrate complex. Okay.
Hey,
Does anybody
Josh,
have any questions on that? Yeah.
could you like visually tell what's going on? Because I don't understand, like high Km implies high Vmax. So that means like to me that the enzyme is liking more of the substrate. So high affinity.
A high
low.
Km, is what you said. A high Km is implying more binding.
Yeah, high Km implies high Vmax, right?
So no,
Higher.
no. Look at this. So Km, let's go back, equals K2 plus K3 over K1, right? So K2, that's leaving your enzyme substrate concentration, right? And K3 is also leaving, right? Do you see that?
Yes.
So that means that this thing doesn't want to stay as an enzyme substrate complex if these numbers are big.
Okay.
Right?
Yes.
Okay. So a high Km gives weak binding, and a low Km gives strong binding. All right. All right, moving on. Okay, Noah Johnson says, what does the strength binding mean exactly? Hopefully this example will clarify that. And if it doesn't, please stop me and we will. Okay, so the turnover number of an enzyme, right, is the number of substrate molecules converted into product, by an enzyme per unit time, right, when the enzyme is fully saturated with the substrate. All right, so in other words, your turnover is going to equal your K3, right? How many of these substrates can turn into product? Okay. And you might see this, as Kcat, right? Okay. And so if you know the concentration of the total number of active sites, which is expressed by ET, right? You have, your total concentration of enzymes. So you know the total concentration of active sites, right? then Vmax gives you the turnover number. Okay, so your maximum velocity is determined by how many enzyme active sites you have, right, times this rate of going from ES to enzyme plus product. All right. And if you rearrange that, right, your Kcat is just your Vmax divided by your total enzyme active sites. And so you get this ratio, right, of Kcat over Km, which is a measure of its catalytic efficiency and enzyme specificity. Okay, so to try to answer your question, Noah, right, what does the strength of binding mean? It means, you know, how specific and how efficient is my enzyme? How well is my enzyme going to perform? That's what the number Km is trying to tell you. Okay. And so let's go through a quick example that will hopefully shine a little light on this. Think about this enzyme, carbonic anhydrase, which is, right, the enzyme that converts CO2 plus H2O into, you know, carbonic acid, right? If you did an experiment, you would get that the Kcat gives you this number, right? 6 times 10 to the 5 substrate molecules converted per second. And you'll get a Km value of, you know, 8 times 10 to the negative 3. And if you, you know, turn them into your Kcat over Km, right, you're going to get a number, you know, 7.5 times 10 to the 7, you know, molar seconds, right? And so for reference, again, the maximum rate of diffusion, you know, onto an enzyme's active site is somewhere between, you know, 10 to the 8 to 10 to the 9, you know, molar seconds. So this number right here then would be considered a very good enzyme, a very efficient enzyme. All right. So let's compare that now to a different enzyme, penicillinase. So somebody says, what quantifies an enzyme as good as and efficient? That you have a high Kcat to Km would make something good and efficient, right? It means that your enzyme binds really well, right? So Km, that's binding, right? Km is the number that is for enzyme binding, you know, how strong this enzyme substrate complex is formed. And Kcat is the same thing as K3, so how quickly you can move it into your enzyme to product. Does that make sense, Jonathan? So it's this ratio that tells you if an enzyme is good and efficient. All right. And so let's go through penicillinase, and hopefully this can, you know, drive home a point. You know, if you did an experiment and you got that the Kcat of, you know, penicillinase is 2 times 10 to the 3 molecules per second, and the Km is 8 times 10 to the negative 5 molar, okay? You'll notice that the Kcat of this is 10 to the 3, and the Kcat of this is 10 to the 5. So you would expect, okay, you know, that's 2 magnitudes lower than, you know, carbonic anhydrase. The Kcat's 2 magnitudes lower. So you might think that penicillinase is actually a worse enzyme than carbonic anhydrase, a less efficient enzyme. However, you'll notice here that the Km is 2 orders of magnitude smaller than the Km for carbonic anhydrase, right? So it turns out that they're actually very similar enzymes. They both have the same amount of efficiency. You guys see that? Right? So, you know, again, it's how strong your enzyme substrate will bind to each other, right? So if this is a very strong bind, it'll make your enzyme more efficient. And if it has a high, you know, Kcat, aka K3, it's going to make your enzyme more efficient. So he says, can you say that again, please, about why they're similar? Okay. Yes, I can. So there's 2 enzymes here, right? Carbonic anhydrase and penicillinase. And you've done your experiment and you've gotten that this has the Kcat, right? Remember, Kcat is just K3 of this number. And you have penicillinase and it has a Kcat of this number. And at first glance, you would think, okay, carbonic anhydrase is a much more efficient enzyme because the KCAT is bigger, right? The K3 is bigger than penicillinase. But that's not the whole picture, because the Km of carbonic anhydrase, right, is 2 matters of, the Km is bigger than the Km of penicillinase, right? Which means that penicillinase has a much stronger enzyme substrate complex. They bind much stronger together. You're more likely to see them in this state, the enzyme plus substrate, okay? And so it's that ratio of KCAT to Km that determines an enzyme deficiency. Okay, really good question. KCAT is the number of active sites. KCAT is K3, not the number of active sites. You see K3 here? KCAT is, let me go back a slide. Right, KCAT is also just K3, right? The number of active sites is K3 times the total amount of, the total number of active sites is just the total number of enzymes, right? And so it's this combination that can determine your velocity max, right? You know, how many enzymes do you have and how quickly they're working will give you the Vmax. So a larger ratio means it's more efficient. Yes. Right? So again, oops, let me see this. Right? Km is ES binding, right? So if Km is small, equals more binding. Right? If KCAT is big, equals more product formation. Okay? And so that ratio will give you this enzyme efficiency. Right? You need both of those to be efficient, because you can't form a product if you don't have an enzyme substrate complex. So they actually need to bind and stick to each other long enough that it can do the chemical reaction. All right. Cool. I know this is a lot. And let's talk about now enzyme kinetics and relating it back to the Lineweaver-Burke plot. And hopefully this will start to all crystallize in your head. Okay. So, right, we've used the Michaelis-Menten equation, and that allows us to graph velocity as a function of substrate concentration. Okay. And we really want to understand Km. This is a very important number, right? It tells us, you know, how well our enzyme substrate complex can be formed. But it's really hard to figure out the value of Km, right? Because, you know, in the real world, you're collecting data points, right? And how are you going to fit this? How are you going to fit this graph to all these data points? It's really hard. It's really challenging. Okay. And so what you do is these people called Lineweaver and Burke, this pair of people, they realize, okay, if we take the reciprocal of the Michaelis-Menten equation, right? So 1 over Vmax, right? Then you get this thing called the double reciprocal plot, or the Lineweaver-Burke plot, which is expressed down here. 1 over Vmax equals Km over Vmax times 1 over substrate plus, you know, 1 over Vmax. Okay. And so now if you take all these data points that you plotted and you plug them into this new equation, the double reciprocal plot, then you get them over here and you can actually form a line through them, right? And so, you know, right, this is Y equals MX plus B. Okay. And so why this is so useful, it's because where this line intercepts the x-axis and the y-axis gives us our Km and our Vmax, right? This means 1 over negative Km, and this means 1 over Vmax, right? Which is really easy to just convert back to your Vmax and your Km. Okay. Are you guys following this so far? Yep. The bottom graph is the reciprocal of the top graph, right? You take this equation and you just put 1 over it and you get this equation. Yeah, and it converts it into y equals mx plus b, right? And if you remember back to your, I don't know, middle school or whatever it is, right? You get the x-intercept and the y-intercept. And that'll actually give you your Km value and your Vmax value. Okay. And so why these are very useful is because if we're studying inhibitors, you know, we can see what type of inhibitor they are based on the Lineweaver-Burke plot. So if we look at, you know, a competitive inhibitor, right? And this is, you know, a substrate that is a competitive inhibitor is a substrate that competes directly, you know, with the normal substrate, you know, for an enzyme binding site, usually reversible. So for example, look at this reaction here. You have succinate, and here's the enzyme succinate dehydrogenase, and it'll turn into fumarate. Okay. It turns out that malonate is a competitive inhibitor of succinate dehydrogenase, and will actually bind into the active site, but you'll get no reaction. No, you're not going to be expected to calculate any of these variables values on the MCAT, but you need to know what they mean. And that's why we're going through this, you know, pretty slowly. You know, it's to understand, you know, what's the significance of Km? What's the significance of Vmax, right? You know, they'll get, and we're going to go through an example question, and, you know, and you'll see how hard it can get, but you need to understand what they mean. Yeah, so competitive inhibitor is competition at the active site. Exactly. Right, so you can imagine, here's your enzyme and here's your enzyme substrate. Your enzyme can bind with the inhibitor, right, I, and form an enzyme inhibitor complex, instead of, you know, what you want to happen is enzyme plus substrate forming the enzyme substrate complex, right? Because this will allow you to go to your product. Okay. Now, you'll notice, if by looking at these graphs, right, that the competitive inhibitor, you know, can be overcome at high substrate concentrations, right? So if you just start feeding more substrate, eventually you're just going to outcompete this I, and everything's going to, you know, pretty much go this way. And, you know, it'll go back. It doesn't alter your Vmax, right? So your Vmax stays the same. Right? And this is why the Lineweaver-Burke plot is so nice. It's because when you plot this on the Lineweaver-Burke plot, right, you'll notice that the y-intercept doesn't change. And because you know the y-intercept corresponds to the Vmax, you know the Vmax doesn't change with a competitive inhibitor. Okay. But when you add a competitive inhibitor, right, the Km, which is on the x-intercept, is going to change. It's actually going to increase, right, to reach a new, you know, level of 1 over V saturation. Okay. So you'll see that this changes down here, right? Km is increasing. What does Km increasing mean? If Km gets bigger, what does that mean? Weaker binding, exactly. You have weaker binding at enzyme substrate complex because you've got this competitive inhibitor right here, right? It's competing for that binding. It's going to bind weaker. Okay. Awesome. All right, let's move on to a non. Yeah. And I mean, ideally, you'll just know now what these parameters mean, right? So if they ever throw these at you, you know, you'll understand it, right? You don't have to memorize these graphs. That's the goal, that you don't have to memorize the graphs. Okay, let's talk about a non-competitive inhibitor. Okay, so this is non-competitive inhibitors. They'll bind, you know, somewhere else, you know, other than the enzyme's active site, right? And non-competitive inhibitors, right? They can either bind the free, and because of that, they can either bind the free enzyme, you know, or they can bind, you know, the enzyme substrate complex. Okay, right? If you have someone wants me to go back. Got it. Cool. Okay. So let me write that down, right? Non-competitive inhibitors, you know, can bind at site other than the active site. Okay. Right. So it can either bind the free enzyme here, or it can bind the enzyme substrate complex here. Okay. And so what does this mean? This means that no matter how much substrate you feed, right, you increase your substrate concentration, you're never going to be able to get up to your Vmax, right? Because the enzyme substrate complex can still bind to the inhibitor, to the non-competitive inhibitor, right? Do you see that? And so what this means is that your Vmax over here is going to decrease. Okay. But your Km will stay the same. Do you guys follow?
Can you explain why the Vmax decreases again, please?
Yes, absolutely. So if your enzyme over here, let's erase these so I can draw on them again. If your enzyme needs to form an enzyme substrate complex to be able to form a product, right? And your Vmax, right, it's just how much product you can form, how quick your K3 is. However, if you introduce an inhibitor, you don't get any product formation. So your inhibitor can bind to the free enzyme and go down here, or your inhibitor can bind to the enzyme substrate complex and go down here, right? So no matter how much substrate you feed it, right? You feed substrate and you just keep increasing the substrate concentration, right? It doesn't matter because your inhibitor can still stop it, your enzyme substrate complex, from going to product. So your Vmax will be decreased, right? For every inhibitor there is, it's going to lower how quickly you can get into your product. Now, your Km doesn't change, right? And that's because you can still form an enzyme substrate complex. Does that make sense? And awesome. And Monica, you said something about an allosteric site. Let's do that in a sec, but that's going to be slightly different. Okay. All right, so this leads to non-competitive mixed inhibition. All right, and that's the same thing as a non-competitive inhibitor. It's just a little bit more confusing because in the real world, right, these rates, right, K here and K here, they're not going to be equal to each other. So depending on which is favored, right, either this is going to be favored, enzyme substrate, enzyme non-competitive inhibitor, or the enzyme substrate complex non-competitive inhibitor, right? There's going to be some sort of preference towards this or this. And based on the preference, right, based on whichever one of these is bigger or smaller, you're going to get just slightly different behaviors, right? In both cases, your Vmax is going to go down, right? Because no matter how much substrate you give it, right, an inhibitor will always block it from going to product. But depending on which one of these rates is preferred, either in this case Ki or K double I, the Km might increase or it might decrease. Okay. So do you see here that like if this rate is much more favored than this rate, so you favor the enzyme and non-competitive inhibitor, it's going to act more like a competitive inhibitor than a non-competitive inhibitor, right? If it favors this one more, then it's going to act more like, your standard non-competitive inhibitor, okay? And so your Km here will increase. Your Km here will decrease, right? And Vmax decreases for both. You guys see that? It again, it depends on this, right? Does your enzyme prefer to bind to just the non-competitive inhibitor, or does it prefer to bind to the enzyme substrate complex with the non-competitive inhibitor? Okay, it just depends, right? Both of these are happening, but there's probably going to be a preference towards one of these versus the other. Okay, and based on that preference, it's going to actually adjust, what the Km actually does, right? Okay, and so if this one is stronger, then the Km is going to decrease because it means that the enzyme substrate is going to have a stronger affinity, right? They're going to bind tighter. Okay, and remember, Km decreasing equals binding tighter. Okay. Okay, now let's talk about the last one, which is an uncompetitive inhibitor. Okay, and so these are inhibitors that will bind to the enzyme substrate complex, and again, prevent product formation, but it'll bind specifically to the enzyme substrate complex. Okay. And so what does that mean, right? If it only binds here, that means our Km is going to decrease again, right? But because we're not forming product, right, our Vmax is also going to decrease. And these are actually fairly rare, but you'll notice that the Lineweaver-Burke plots will form these parallel lines, right? And that's because, for every inhibitor, it'll bind the enzyme substrate complex and push it that way, right? All right. Okay. Now we're going to touch on this quickly. There's something called allosteric regulation, right? And that's when, enzymes will regulate their efficiency based on different allosteric effectors, right? Or covalent modifications. So you could imagine an enzyme is, modified by like a phosphate or something binds to the enzyme. And based on the binding, the enzyme, might change shape slightly, and it might make it either more or less efficient. And so you'll get these, what is this, sigmoidal graphs, right? Or here's your velocity, and here's your substrate concentration. Right, and depending if it's a positive or a negative effector, it'll move it this way, right? Okay. And the takeaway for these is these allosteric regulation does not follow Michaelis-Menten kinetics, right? Because it shows cooperativity between amount of binding. And someone says, how much overtime do you think? We're going to go very overtime. This is taking, quite some time, but it's good. It's good to go slow and, understand this stuff. And we can rush through some things that, don't need to be explained too much. But I do think it's good to do practice questions, right? Which we're actually going to do right now. Okay. So someone says, so, okay, this is very, I would actually recommend reading about that. So someone asked an amazing question. So when it binds to the ES complex, is that not allosteric? It's an allosteric site, but it doesn't follow the Michaelis-Menten kinetics, right? So you can imagine a protein, so the difference between allosteric regulation and non-competitive inhibition is that on allosteric regulation, the enzyme is actually going to you can imagine like an enzyme changing shape, and that's going to either make it more or less efficient, right? Versus a non-competitive inhibitor, which when it binds, it doesn't allow any product to form. Does that make sense? So like some things you can, right? Some things like hemoglobin, right? How well it binds to oxygen or not is allosteric regulation, because, depending on the concentration, depending on how many, whatever, 3 BPGs, you'll get into this later, bind, it'll either have a higher or lower affinity for oxygen, right? Versus a non-competitive inhibitor, when it binds to, a site other than the activin, it's just not going to allow any product to form. Michaela, does that make sense? Okay. And we'll talk, we'll talk about allosteric regulation, later. There's like the classic hemoglobin, right? How well it can carry oxygen based on, other things that bind to it and activate it or deactivate it, right? But you don't get that turn on off, right? You get these sigmoidal curves, right? Yeah, I think that's a good idea. At 4 p.m., let's take a quick 5 for all those who are going to stay. For everyone else, we might have to do a mop-up session if you must leave. I totally get it. But let's do this question and then maybe take a quick 5. Okay, so I'll give you guys another 5 minutes and then start guessing. Yeah, and hey man, we're gotta dive in. We got information overload here. And again, with this stuff, these are, all these concepts are very, it takes a while to digest them, right? So go back through the slides later. Office hours, right? To clear up any of these equations or graphs, or what does Km mean, or all that. It's a great time to come back and do it. Let's go over this stuff. Okay, so the first question, right? So this is going to feel like the MCAT, guys. This is an MCAT passage. So lots of, molecule names, lots of technical things getting thrown at us. Okay, so we got a mix between competitive inhibitor and non-competitive inhibitor. So does anybody want to say why it's one or the other?
I said it was B because in the passage it said that it said it cannot be further metabolized by hexose isomerase, and it's
Which
trapped
one can't?
into, 2D or 2-deoxyglucose, 2DG. So
Okay, so you're saying B because 2DG cannot be phosphorylated by which enzyme? Because there's two enzymes in this passage, right? We're talking about phosphorylated by hexokinase, right? And then cannot be further metabolized by phosphohexose isomerase. So you have to be very careful here. There's two enzymes, okay? And I'm going to say that down here, right? It literally says it directly in two areas, by its natural counterpart, glucose. Okay. And the inhibition of this rate limiting step by 2DG, leading to inhibition of glycolysis step at the step of phosphorylation of glucose by hexokinase. Okay. So I'm going to argue that 2DG acts as a competitive inhibitor. The answer is A, by inhibiting hexokinase, right? Nice, nice, very good. Okay. So I can see why you put non-competitive inhibitor, but in the passage it specifically says that 2DG is inhibited by that first enzyme hexokinase. Okay. And he says, why is it not B? Actually, I think I put it here, right? So it turns out that, so you have to, here's the first step, okay? So here's glycolysis, which we're going to get into in a sec, but just to have it here. Glucose goes to glucose 6-phosphate by hexokinase, right? And then glucose 6-phosphate goes to fructose 6-phosphate by isomerase. Okay. And so in the passage, it says that 2DG acts as an inhibitor from glucose to glucose 6-phosphate. It acts as an inhibitor of hexokinase, right? So essentially what's happening is 2DG gets in there, right? And prevents glucose from being turned into glucose 6-phosphate. Cool. So let's go back here. Okay. And so, what does this mean, right? This means it's a competitive inhibitor. This means that the Km will increase, right? But the Vmax will stay the same because you can always outcompete it by adding more glucose, which is why down here, this is like a second hint, right? The effectiveness of 2DG is significantly affected by the presence of its natural counterpart glucose, right? So if you increase the substrate concentration, right, you can actually get back to your Vmax. So that would be the other telltale that it's a competitive inhibitor. Okay, let's go down to question number 4. And this is just to say this is a trick question, okay? Because if you read this, based on the passage, which of the following observations can be made about the conversion of glucose 6-phosphate to fructose 6-phosphate by phosphohexose isomerase? Okay, so this is actually testing your ability to picture glycolysis and the reactions that occur before and after, right? So we're not actually talking about, oops, if we go back here, we're now talking about this enzyme. What happens to this enzyme, right? And we know that the inhibition was occurring at this step, right, at hexokinase. Oops. Right, so does the Km increase? Nope, it doesn't affect the Km because it's not acting as an inhibitor there, right? What about Vmax? Does that decrease? Right, Vmax is gonna, stay the same, right? There might be a lower, reaction velocity because there's less total substrate, but the Vmax isn't going to be changed. So we can cross that out. Okay. And, between these two, right, glucose 6-phosphate or fructose 6-phosphate increasing, the answer is going to be actually C, glucose 6-phosphate concentration decreases. Why does the glucose 6-phosphate concentration decrease? Right, oops. There we go. Because if we're feeding a mixture of glucose in a 1-to-1 ratio with 2-deoxyglucose, you're going to have an overall less amount of glucose, right? So you're going to have a less amount of glucose 6 phosphate. Does that make sense? You're going to end up with 2-deoxy glucose phosphate, and you're going to end up with glucose 6 phosphate. And why would D be wrong then? Because is the fructose 6 phosphate concentration going to increase? Is the glucose being converted to fructose in this scenario? Yes, the glucose 6 phosphate is going to be converted to fructose in this scenario. Correct. Right? 2-DG is not going to be converted into fructose in this scenario. Right? 2-DG gets stuck. Right? As you can see down here. Right? Hexokinase will work on it. You'll get 2-DG6 phosphate, but you're not going to get 2-DG 6 or 2-DG6 fructose, right? Or whatever, the 2-deoxy fructose. You're not going to have that. Okay. Does that make sense? Do you guys see this? Okay, awesome. Cool. Yeah, and so this is, again, this is really hard. This is really hard, right? But they're giving you an information overload. And the better you know glycolysis, the quicker you're going to be able to solve this passage, right? Instead of having to try to translate what these enzymes are doing, you're going to kind of have it pictured in your head already going into the passage, and you'll be able to much quicker get to an answer. All right. Yeah. Okay, it's 4 o'clock. Perfect. Do you guys want to take a quick 5-minute break? Okay, yeah, let's do it. Well, how about we come back here? Let's make it a little longer. 5 minutes doesn't do anything. We'll make it till 4. How about split the difference? 4:08. We'll start it up again. And yeah, I can go back to the last slide. So anybody has any questions? If you guys are just totally burnt out and done and just need to just walk out of here, I don't mind figuring out a way to cover, maybe not like a lecture slot, but we'll meet and we'll specifically go over this next part of the lecture. Okay, if I'll be willing to do that. And I can't answer that. Depends how many questions I get for how much longer this goes. But I mean, we got to talk about metabolic components, which we'll kind of maybe brush over. And then we're going to talk about glycolysis, which we're not going to brush over. I could send you the slides with writing on them, but I mean, the problem is if you look at my slides with writing on them, it's kind of messy, right? Might be better to just do another mop-up lecture. I think we'll just do another mop-up lecture. It's good. This stuff is so dense. It'll be good to do that. But I will keep going, right? And try to do this before. Try to get this done by 5. Yeah, and I totally understand. If you guys got to bounce, but we'll do it. Anyways, somebody have a question?
I was just wondering, it seems like the bio lectures are pretty dense and heavy and we don't get through them in the 2 hours. Would it be possible to maybe like add another lecture for them, like consistently, or?
I think that's a good idea. I think what needs to happen is, yeah, there needs to be at least one lecture between the 2, right? So like last lecture, should have been split up into 1.5, and this should be split up into 1.5. And honestly, the metabolism lecture should probably be split up into 1.5. The other ones will be a little better. I mean, this stuff is just so, it's just so, I mean, this is, think about it. We're trying to cover an entire semester of biochemistry in 2 lectures.
Yeah, so when do you think we would be able to start adding another lecture?
I don't know. I'll have to look at the schedule, you know, and that's just, we'll just have to be flexible. And I appreciate the flexibility, right? I will try to give you guys more than 1 day advance. The problem with, you needed structure and function to be able to go into this. They kind of go in a sequential order. And so I did not want to just jump into this lecture without doing the structure and function lecture. That would have been a big mistake. And someone says there was inconsistencies in the assignment. What was the inconsistency? For the bio? I don't think so. What's in those assignments is what you should do, right? So like this, we're trying to cover section 7, which is the metabolic components section. Right? Really? I don't. What can someone give me like a specific? What does it say to do that we shouldn't be doing? Not to pry, just I looked at it earlier today and it looked totally right, at least for this lecture.
I know like sometimes it seems like for, well,
Yeah,
yeah,
is that not right? That's what it says to do. Sorry, I'm not trying to be, I'm just trying to say, like, does anybody disagree with that? Read page 105 to 127. Right, enzyme class handout. Like that's literally what we've just been covering right now. Right? Monica, am I wrong? Oh, no worries. It happens. I know one person has a different version of the book, or maybe 2, which might be different, but like, I mean, just the homework assignments should correspond roughly to it. It's never perfect, but like if you were to start flipping through the chapter, you'll see, hey, we're talking about enzyme kinetics, right? Yeah. I mean, and yeah, if you ever feel like there's a discrepancy, let me know so I can fix it. Someone said that earlier today too, so clearly there's multiple people confused, but I looked at it and it looked fine. So that's why I'm confused. Right. And so metabolic components, right? If you're like, wait, this doesn't make sense, maybe open the book and start reading metabolic components and realizing, you know, that it's going through a bunch of enzyme kinetics, right? And if the titles, they might be slightly different, but like it's the same content. Right. Right. Cool. Right, and that's going to be especially the case when like psych and soc start happening, right? It's not going to line up perfectly, but you know, you just got to be on that topic. Yeah, this is metabolic components, right? If you start flipping through the metabolic components chapter, if you've done the reading beforehand, you'll see that it's very, I mean, it's all that stuff. The only thing that would be different is just because we're trying to get ahead on glycolysis now.
I also had another question, Josh.
Yeah, of course.
I know you mentioned that something happened with the biology professor in the beginning. Do you know if she will end up coming or are you gonna take her place?
I'm going to take her place. One of the person who was doing genetic info and expression might pick up one of those lectures, but if not, I'll just do them.
Okay, thanks.
Yeah, I hope that's okay.
Yeah, I was just wondering if anything else would happen with that because we didn't hear anything else.
Yeah, I'm still trying to find out. I'm a little nervous. This is people who don't show up for work is like usually that means something bad happens. It's like the first sign of something bad happened. So I don't know. I hope I find out. That's all I can say. Cool. All right, let's try to keep going. Hopefully you guys have been able to, have a sip of water, walk around, get some fresh air. So now we're going to be talking about metabolic components. Okay. And first we're going to talk about, how different things will affect enzymes, right? So pH, as I alluded to in the last lecture, is incredibly important for the efficiency of an enzyme. All right. And for any given enzyme, right, there's usually an optimal pH range, and it's usually pretty narrow. Okay. And so, down here, right, catalysis, right? So if it's an enzyme, whether it can, break a bond, put on a bond, whatever it is, the catalysis, right, it's going to depend greatly on the protonation state of the functional group, right? And when I say functional group, I mean the residues, right? So here's amino acid residues that are in the active site of the enzyme. Okay. And so, for in an active site, for it to act as a good nucleophile, the dissociating proton, so the proton that's leaving the residue, right? It must leave, so it leaves a reactive, chemical species, generally nitrogen, oxygen, or sulfur. Okay. Right. So what that means is that the pKa of the dissociable proton, right, from my pKa's, has to be less than the pH of the active site, right? So good examples would be, your acidic residues, your histidine, right, at a pH of 7.4, physiological pH. All right. And so the opposite of that, right, the pK of the electrophile, has to be greater than the pH of the active site. So you need to have some sort of charge. And this is going to be seen mostly on your basic residues, right? Your lysine, your arginine. All right. And then something called general acid-base catalysis, your functional group has to have a pK similar to the pH of the active site. And we'll talk about this a little bit, we're talking about chymotrypsin, right? But that's why histidine is such a special amino acid residue, right? And that's because its pKa of its functional group is close to physiological pH, so it can both act as an acid and a base, depending on the certain situation. Can either accept electrons or, donate them, right? So does this make sense to you guys at all? Right, so like here's glutamate up here. Okay, and the pKa is equal to 4.2. Right, so therefore, at a pH of 7.4, it can actually act as a base, right? And it can grab a hydrogen. Okay. Similarly, histamine, which is this one, can also act as a base at pH of 7.4, right? And it can grab a hydrogen, all right? The opposite would be the basic amino acids, right? So like lysine, this is lysine down here, right? Its pKa is around 10.0. So at physiological pH, it'll actually be protonated, okay? And therefore it can act as an electrophile, right? And form a bond, right? By grabbing an electron pair. So it can act, right? Right. So remember, like an electrophile, that's something that accepts electrons to form a bond, you know, and a nucleophile, which is something that will donate electrons to form a bond. Okay. And so temperature also plays a huge role. Temperature also has a huge effect on, you know, the velocity of a reaction. Okay. And so you can imagine you have an increased temperature, you're actually going to be able to speed up the velocity of a reaction. Okay. You know, you have molecules, the higher temperature, they're banging into each other more. So things, you know, move much faster. Right now, you know, obviously we're to a certain point, right? At a certain point, it's too hot and you'll actually, you know, denature the enzyme, in which case the velocity will very quickly decrease. The enzyme won't be active anymore. All right. And so, you know, you can even see this, like, have you guys like ever been like outside and it's like your hands are freezing? Okay. Your body actually has these like cold adapted enzymes that can help, you know, catalyze reactions in colder conditions. So that's why like your hands will start to hurt if you've been in the snow for forever, you go ice climbing or something like that. Right. You know, and if you can even compare enzymes between species. So like, you know, mammals that have, you know, lived in the cold, like polar bears or woolly mammoths, right, that have lived in the cold for a very long time, you'll actually notice that if you compare their genomes, their enzymes are cold adapted. So their actual optimal temperature has been shifted over one, you know. Have you guys heard of that? What's the guy, the geneticist from Harvard, George Church? He's actually trying to bring back the woolly mammoth. They've, you know, obtained woolly mammoth DNA, and they've actually sequenced it, and they've found genes in it that are cold adapted. And so they're trying to create this elephant woolly mammoth hybrid. And his goal is to introduce the I don't know, pseudo woolly mammoth into the Arctic tundra, right, into the boreal forests of like Russia. And they've actually started this program, you know, that's expected to have its first woolly mammoth in like 10, 15 years, or, you know, pseudo woolly mammoth. That's adapted to the cold. It has all these enzymes. So it's essentially like a cold elephant. Yeah, exactly. It's essentially, it's going to be this like elephant that's now adapted to the cold using like woolly mammoth genetics. And the idea is that these woolly mammoths, if they can populate boreal forest, can knock down a lot of those trees. And if you knock down a bunch of trees, then grass grows. And it turns out that certain, depending on where it is, aka the Arctic, grasslands can actually sequester more carbon dioxide than a tree can, right? Just because of the growth, you know, storing it all in the soil, you get actually a lot more carbon sequester, you know, with grasslands up north. So, you know, that's his justification for trying to bring back a woolly mammoth. Crazy. Anyway, tangent, but it's cool. Okay, so now when you're talking about inside an enzyme pocket, right? So the active site of an enzyme, you get these microenvironments, and you're going to have these electrostatic effects that will alter, you know, the pKa and alter the function of residues within the active pocket. Okay, so what happens when you bring, you know, two negatively charged residues, in close proximity of each other, right? So like here's an enzyme pocket. You have, aspartate and glutamate, two negatively charged amino acids. As we were saying earlier today, this doesn't like to happen. This is bad energetically. This is unfavorable. They don't want to have two negative charges next to each other. So one will actually increase the pKa of the other in this microenvironment, and thereby protonating one, or allowing the other to be more easily protonated, right? To balance out those negative electrostatic effects. Okay, so yes, great. The pKa would increase. Nice, right? So pKa increases in residue, right? Okay, and then what about two positive ones, right? It's going to be the exact opposite, right? They hate this, electrostatic positive charge effects. So the pKa is actually going to go down in one of the residues, right? Allowing it to be, deprotonated. Okay. And so this is very fundamental in why different combinations of amino acids have very different and specific, reactions, right? The size of the enzymatic pocket will allow certain things to fit or not fit. And then just the way the interactions between the actual residues in the active site, right, will allow them to be better nucleophiles or better electrophiles. Okay. And so if you have, two that are opposite, this is actually energetically favorable. And so it'll actually work together to maintain that positive and negative charge. Okay. I think I actually wrote this out, or maybe not. So, how does it affect pK again? Here, let's go back. Right, so this will, let me just write this out. So two negative charges will increase pKa of one of the species, right? pKa of the other group, right, and thereby protonating it. Right, and this will decrease the pKa in the microenvironment of the other one, and thereby deprotonating. Right, and this one will it will push the pKa's to the other extremes, right? So it'll maintain, right, slash, right, so the positively charged one will decrease, right? So this pKa you got that? So they'll actually act in complement to each other. So this thing's pKa will increase, and this one's positive charge, the pKa will increase, negative charge, the pKa will decrease, right? Because that's more energetically favorable. Yeah, and just one of the groups, they do it to each other, right? But just one of the groups is going to get the proton, right? Because, they both still want to be negative, right? Because they both, in this case, still have a low pKa, but they're just going to influence each other enough that one of them is going to end up grabbing a proton to make this more, to reduce the electrostatic repulsions. Okay.
Can I ask a question really quickly about
Yeah,
the
of course.
pH for,
Yeah.
I'm not sure if you mentioned this already, but when I think of saying increasing pK doesn't really make sense to me, but protonating it does. But if you're protonating one of them, wouldn't the pH be decreasing? Is that
The pH
the
of
correct
the pKa.
The pH, not the pKa.
Wait, say that again. It's a good question. Just can you say it again?
Yeah, I'm just having a hard time understanding the idea of increasing and decreasing pK. It makes more sense to me to think more in terms of pH. Because when I see the word like protonating it, and that just means that like there's a lot of like hydrogens within the solution, so that means there's a decrease in pH. Is that the correct way to look at this?
I would say no, but I see what you're saying, right? So let's go back here just for a second, just because we're looking at amino acids, they're drawn out. Right, so this amino acid, the functional group on this amino acid, right? On glutamate, right? It's in the deprotonated state. And the other one would be if you have the same amino acid, and I'm just going to simplify it here, right? In the protonated state.
Right.
Okay. And this thing's going to have a pKa of around 4. pKa, I don't know, 4.2, I think I said, right? And so what that means is at a pH of 4.2, so if pH equals 4.2, then you're going to have half of the molecules are going to be protonated and half of the molecules are going to be deprotonated.
Right.
Right. So if we're at a pH and physiological pH of pH equals 7.4, what does that mean?
That it's deprotonated.
Yep, exactly. That it's deprotonated. Perfect. Right.
Right.
And so think of it kind of as like a sliding scale, you know, the higher the pH is, the more species are going to be deprotonated. The lower the pH is, the more species are going to be protonated.
Right, because there's more hydrogen ions within like the whatever solution that you're in.
Exactly.
Right, that makes total sense. So that like makes my question what I'm asking, where you're saying an increase in pKa, but wouldn't there be a decrease in pH because you're protonating the molecule or the amino acid, right?
Gotcha. And so adjusting the pKa is just adjusting how likely it is to associate or to the protonate or deprotonate, right? So it's just, if you were to adjust this pKa to a slightly, you know, higher pKa, let's say pKa 6.0, right? And we're at a pH of 7.4, what does that mean? Right? So went from 4.2 to 6.0, and now we're at a pH of 7.4, right?
Right.
Are there going to be more or less protonated species?
I see. I mean, it's still deprotonated, but not to a like significant degree.
The population of each of these two species changes based on the pKa, right? At that pKa. Yeah, really good question.
So why would you look at it with pKa instead of pH?
Because the pKa is describing, you know, the pH at which these species are in equal concentrations, right? It's just the pKa is literally describing at this pH, you have 50% in this one and 50% in this one, right? So if you start to increase or decrease your pH, you're going to see that that equilibrium shifts to one way or the other. Yes, and it's exactly right. It reminds you of titrations.
Got it. Okay, thank you.
Yeah, really good question. Okay. All right, I think we were here, enzyme mechanisms. Yeah, so these are like for anybody who's taken, I don't know, hopefully you've taken, you know, biochemistry classes, right? They go over, you know, usually just one or maybe all 4 of these, you know, classic enzyme mechanisms, right? And you just need to understand like the principles of an enzyme mechanism, which we've been, you know, going over. And so we'll just super quickly go through the chemotrypsin mechanism, at least the first step to try to solidify all this. It's also in your book, and you can also always just look up these if you want like more, but it's just, you know, how these microenvironments allow an active site to work, okay? And so if we look at chemotrypsin, right, we're in the active site, so we have, you know, a serine residue, a histidine residue, and an aspartate residue, all in that little pocket. So they're all very close to each other, right? So you're going to get these electrostatic effects of each other, okay? And serine's pKa is usually around a pKa of 13, right? So it really, really wants to hold on to that hydrogen, okay? Histidine's pKa, right? is 6.0, so it's usually unprotonated at a physiological pH of 7.4. So I think it's something like, it's usually like 96% unprotonated at, or at pH 7.4, right? However, aspartate, right, it can act as a base and it can interact with histidine to make histidine an even stronger base, okay? So it can the aspartate, right, this negative charge can act as a base and try to grab this hydrogen from histidine, right? Which in turn turns histidine into something that can act as an even stronger base, and strong enough that it might even grab this hydrogen from this serine, right? And so what that does is it leaves this serine as a very, very, it's now, it's called an alkoxide, right? Which is a very strong base, and it's a great nucleophile, right? So this thing, right, is going to go want to find like a reactive center. So, a C-double-O bond, a carbonyl bond, right? This is going to looking for it. So if you have a substrate coming around and floating in here, this thing is going to want to go attack it, right? Which we can see here, right? So you have this, and here's your substrate. In this case, let me go back one. Right, chymotrypsin, what it does is it hydrolyzes ester or peptide bonds, right? So you can see this amino acid right here. This substrate comes into the pocket, and now we have this activated serine, and it's going to want to go attack this nucleophile. It's going to bind. And so what you do is you get this, this very unstable tetrahedral transition state down here, right? And this is why an enzyme lowers the activation energy, because an enzyme, right, can make this more intermediate. It can stabilize it more, and stabilize it enough that the enzyme can proceed, right, and actually cleave it off with these reactions, right? So right here, you can see that now the peptide bond here can be cleaved, and you're playing this big game of electron shuffling, between the different species in the enzymatic pocket, right? And down here, you can see that your amino product can actually leave. Okay. Right, and this is just technical. Obviously, you don't need to know this, right? But this is how it goes back to its its natural resting state, where the enzyme is not changed or altered in any way, right? Okay, right. So the histidine here can act as a base, right, and grab a hydrogen from the water, right, which makes the water act as a nucleophile, and then attacks the carbonyl bond, on this intermediate molecule, right, which gives you another, transient unstable intermediate, right? And similarly, right, histidine can now act as an acid, right? And serine can act as a base, right? It can grab the hydrogen, and then now you're back here with where you started in the enzymatic pocket, and then the other product can leave, the acid product. Okay. Right, so we went through that pretty quick, but the big takeaway here is different enzymatic pockets will have different interactions between the active residues that allow things to be good nucleophiles and good electrophiles, and so it can go attack reactive carbonyl centers. Okay, and so you can see if the pH was messed up, right? If the pH was too high or too low, this enzyme is not going to be active anymore, right? Because it's not going to be able to turn serine into, this nucleophile, right? Yeah, great. The main takeaway is that different combinations of residues will have different effects on each other in the enzymatic pocket, in the active site of an enzyme, okay? So depending on the enzyme, it'll have a different combination of amino acid residues that have different electrostatic effects on themselves that either turn things into electrophiles or nucleophiles, or, you know, the electron sinks, electron donors, right? So when a little, let's go back one. Well, let's go down one, right? So when you have when you have some sort of substrate get near it, this serine, which is normally in this example protonated, is actually unprotonated because these things pull that proton off and it'll become active and it can attack the substrate. Does that help, Noah? Awesome. Okay. And we're going to brush through this super fast because there's a really good handout on it that's actually better than what I have here in the lecture. But for the MCAT, please, 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 please know your enzyme classes. Okay, there's 6 of them, EC1 to EC6. And you should be able to identify, if you were given a reaction, what enzyme class it is. And I can't tell you how easy these free points are on the MCAT. They're asked, you know, it's the same thing with, like, you know, PKs and stuff, right? The enzyme classes, just, you know, I don't want to tell you to memorize too much stuff. These are something I want you to memorize, right? And there's a handout on the portal that's really good. It goes through EC1 through EC6. And I think the best way to test yourself to see if you know these, which we're actually going to do, is each enzyme in glycolysis, or even the citric acid cycle, if you want bonus points, you should be able to tell yourself what enzyme class it is, right? So the 6 enzyme classes are oxidoreductase, right, which is the catalyzing oxidation reduction reactions, right? So this is, you know, you transfer, you know, electrons from a donor to an acceptor. I can actually draw out the general reaction. There's a couple examples below, right? A plus B goes to A plus B. However, one is reduced, one is oxidized, one is oxidized, one is reduced. Okay. Okay, and again, so with every enzyme class, the main enzyme class, there's all these subclasses. And this is why it becomes challenging, right? Because you'll get an enzyme name, you'll be reading a passage and it'll say the blah, blah, blah dehydrogenase, you know, did this reaction, right? This is a subclass of the oxidoreductase general class, right? And so you need to know what the general class is, all right? Let's get through these pretty quickly just because you can read that handout. Transferase, okay, that's, you know, transferring a functional group from one chemical to the next chemical, right? So if you want me to do the general reaction, right, this is if you have, you know, your chemical A and it's got this functional group R, and you add this chemical B, right, then you're going to get A plus B with R. I would memorize the subclasses. I mean, that would be bonus points. I mean, you guys have so much information to do. I think an absolute no is to know the enzyme class, right? Because from what I've seen and what I've heard is, you know, you'll get a question and it's like, it'll show you a reaction. So how you would see this on the MCAT in its most basic form would be, you know, there's a passage and it shows you, you know, in one of the figures, it shows you this. And then one of the questions says, you know, what class of enzyme is it? Right? It's as simple as that. And then it'll have, you know, A, B, C, D, right? Transferase. Gosh, my handwriting, I'm going too fast. Right? Transferase, right? Isomerase, oxidoreductase, right, and so on and so forth. You have to like literally pick it out of the hat. Subclasses, there's too many subclasses. I don't think they would test you on subclasses, but they might use the subclass in the passage to describe the enzyme. And then they might ask you what the enzyme class is. And so if you knew the subclasses, you could super quickly get to this. But otherwise, you should just be able to look at a chemical and see what happens, right? So like here, or here's a simpler one, right? You took ATP and you added a phosphate group to this protein. So a protein kinase, which is actually a subclass, you will end up just knowing it's transferring a phosphate group from a ATP molecule. That's what a kinase is. It's a subclass of the transferase. Okay. All right. Hydrolysis. We'll just go super quick just because I'm just reading these slides essentially. Right, catalyze the cleavage of single bonds by the addition of water, right? So your general reaction is, AR plus water goes to okay. Lyase, it's catalyzing the non-hydraulic cleavage of single bonds. Oh, sorry. Yeah, if you're trying to get this down, I'm sorry, I'm going fast. But yeah, the general reaction is probably good. And again, I think, me just telling you what it does isn't going to be the effective way for you to learn it. The effective way for you to learn it is to read the handout and then go look at reactions and see what you think it is. Okay. And so, for that reason, I'm just going to put it on here for like 10 seconds, right? Lyases, the non-hydraulic cleavage of single bonds. Okay. And I guess I could give you the general reaction for this. So this is like if you have functional group attached to A, attached to B, attached to a functional group, right? And then you have your enzyme, right? And then you get, like A having a double bond to B plus the two functional groups. Functional groups bonded, right? So it cleaved single bonds and formed a double bond, right? Cleaved and formed a double bond. Oh, and this is, let me erase this. That's a single bond. Cool. Isomerases. Okay, this is moving a functional group from one position to another within the same molecule. Okay. So the key is that it doesn't alter the atomic composition, right? So you don't lose anything on the molecule, right? It just moves a functional group from point A to point B. So like a general reaction would be if you have a molecule, A, B, C, right? And then you were to rearrange it so it's B, A, C. Right? You didn't change the molecule, you just move a functional group around. Yeah, same composition, just different location. So like the classic would be, we'll see a classic one in when we talk about glycolysis. Right, and then the last one is ligase. So this is the formation of new carbon-carbon, carbon-oxygen, carbon-nitrogen, carbon-sulfur bonds by coupling that formation with the hydrolysis of ATP. Right? So ATP is involved. So you can imagine like A and B plus ATP goes to A is now bonded to B plus ADP, right? So we had to burn some energy to do it. Oh, and plus PI, balanced equation. All right. So now let's just jump right into glycolysis. And with all these metabolic pathways that we're going to talk about, and I'll get into this in the next lecture, but you absolutely must know the net reactions of each pathway, right? So what does glycolysis produce? What does TCA cycle produce? What does pentose phosphate pathway produce? What is, right? And you should honestly know what the actual reaction does and how questions are going to be asked on the MCAT in regards to any of the metabolic pathways is they're usually going to present you a single reaction within a pathway, right? So it's going to talk about some sort of regulation site, within the cell somewhere, and it's going to be like it's regulated at, the citrate turning into isocitrate, just to pull one out of a hat. And so it'll give you like it'll zoom in on a particular reaction, and then it might ask you, what happens to the cell, right? And you have to kind of, from that single reaction, zoom out and say, okay, well, that's going to, slow down the TCA cell, and that'll, reduce the amount of NADH, and that'll, reduce the total ATP output by ATP phosphorylase, right? Or ATP synthase. Okay. So the more comfortable you are with the metabolic pathways as a whole, when you get a question and it's like zoomed in on a specific reaction, right? If you know where that reaction is coming from, you know how it's going to affect metabolism on a much grander scale. Okay. Good question. So it would be forward only, right? And that's because, well, that's not necessarily true. You're burning ATP, right? But sometimes you can burn ATP, and the product you make from the burnt ATP can actually be reversed, and it's going to have the same amount of delta G, in which case it could actually be reversible. So actually a good case would be down here, right? Right. So this burns ATP, and so this is most likely not going to go this way, right? However, this species that you create, this thing is also probably pretty unstable, and it could probably go back just as equally as it was to go forward, right? So you burned an ATP, but you could actually recapture that ATP, going the other way, probably pretty easily. Right, so it depends. And actually, there'll be an example of that now that I think of it in glycolysis. Okay. Okay, so for glycolysis, right, here's the overview. Okay, so the point is, it's the metabolic pathway. It's actually like the very center of metabolism, in which energy is released from glucose, and it's captured in the form of ATP. So it's captured as chemical energy by creating these ATP molecules. Okay. Now, glycolysis does not require oxygen, which we're going to touch on later when we talk about, fermentation versus oxidative phosphorylation. Okay. But the overall reaction is you take one glucose and you turn it into two pyruvates. Okay. And glucose to pyruvate, the delta G naught prime of this is going to equal 133 kilojoules per mole, right? And if you go from two ADPs plus two NADs plus two phosphates, to 2 ATPs to 2 NADHs plus 2 waters. Oh, sorry, this is a negative. The delta G of this is going to be positive 61 kilojoules per mole. So generating this ATP is not favorable unless you pair it with the breakdown of glucose going to pyruvate. And so the net delta G is going to equal, negative 2 kilojoules per mole. No, you're not expected to know the delta G naughts, but you should, kind of like that first question I asked you guys, right? Which one of these will release the most energy? That is fair game, right? You don't need to know the actual number, but you should know that something is going to be favorable or unfavorable, right? Okay. So like knowing that glucose to pyruvate is favorable because that's what can recoup all this chemical energy in ATP, right? So this reaction right here is unfavorable normally. So if you just had like, ADP floating around, right? It's going to be very unfavorable to go into ATP, right? It's a positive delta G naught prime. However, glucose to pyruvates has a pretty big negative delta G naught prime. And so if you couple these reactions together, which is what glycolysis is, you get this overall reaction of glucose plus ADP plus NAD plus your phosphates, generating 2 pyruvates, a net gain of 2 ATPs, a net gain of 2 NADHs, and some water, right? Okay. Yeah, so this is your starting product. This is your ending product, right? So we're going from a 6-carbon molecule to 2 3-carbon molecules. And in the process, we gain a net 2 ATPs and all this. Glycolysis has 10 enzymatic steps. And several of them, which we will go over, are very, very regulated, right? 10 different enzymes. Okay. And so here's an overview of glycolysis. Okay. Right. Taking glucose, you're adding phosphate, you're turning glucose into fructose, you're adding another phosphate, right? You're splitting up the 6-carbon into 2 3-carbon molecules. You're putting on another phosphate to the 3-carbon molecules. You're now breaking off that phosphate and capturing it as energy. You're this is an isomerase reaction, right? We're moving this to here to make it more energetically favorable, to turn it into this molecule, which is a high energy molecule. And I'm going fast right now. We're going to go into every single step, right? Generate more chemical energy, right? And then you're left at pyruvate, right? And so what I want to do is I want to look at each of these reactions, 1, 2 through 10, individually and give you like highlights of them. And I'm going to put them like kind of isolated by themselves just because I want you to get into the habit of starting to visualize what's before and what's after a reaction that's zoomed in, right? And so the important things are, for step 1, right, glucose to glucose 6-phosphate, right? What enzyme is this? What enzyme class? This is a transferase, right? The name is, it's a hexokinase specifically. That's what the enzyme is called. Hexokinase is a transferase, right? And a kinase is transferring the terminal phosphate of an ATP, and it's actually attaching it right here onto the 6-carbon of glucose, turning it into glucose 6-phosphate. Okay, so why? So good question. Do you recommend knowing the overview in terms of steps with structures of each molecule, or just the steps and what happens in each? I would know both. So glycolysis and TCA, I know them inside and out. I mean, I'd go as far as saying memorize them. Which which just comes from doing practice problems and reading it and, you know, drawing it out. The best thing to do is draw these out yourselves, and you'll actually remember things pretty quickly. Right. And you'll see, right, and I'm going to try to explain to you why each of these steps are important and what it means, you know, from a physiological standpoint in your cells. Okay, so for example, you know, step one, right? What's so important about this step? There's two things, actually. The first step of this is that it's a commitment step. Right, you're burning an ATP, you're investing energy to do this, okay? And yes, it's not reversible, right? You burn that energy, you can't get that back, right? Until you've finished glycolysis, okay? You've invested one ATP. All right, why else is this important? So if you remember yesterday, when we're talking about molecules being able to pass membranes, or sorry, Tuesday, molecules being able to pass membranes, right? Molecules that are charged are going to have a much harder time passing a membrane than a molecule that's uncharged, right? And so it turns out, right, I'm going to draw, you know, a cellular membrane right here down the middle, right? Glucose, right, can actually be transported into the cell. So here's in, here's out, right, by, you know, one of these transmembrane proteins specific for glucose. And now once you get glucose into the cell, glucose can still leave the cell unless you phosphorylate glucose. Now all of a sudden there's this big negative charge and glucose can no longer leave, or glucose 6-phosphate, sorry, can no longer leave the cell. Okay, so this is a really important step. Step one is glucose gets trapped in the cell. Okay. And before we keep going, let's do another quick question. Give you guys 4 minutes, 30 seconds to do this. This is going to be a good example of a hard MCAT style question, right? Okay, cool. Let's do it. So for A, we've got, for 5, we've got a bunch of A's. I think I got only A's. Nice. That's great. So the answer to number 1 is A. Great. And so that's just by knowing your Km values, right? So if you have a high Km value, it means that glucokinase is going to have a weaker binding to glucose. So it's going to be only active when there's a high enough concentration that it can actually, make that reactants to products go through, right? And then it says here that the animal isozymes are allosterically inhibited by the product, glucose 6-phosphate. Okay. So at higher concentrations, all those other isozymes are going to be inhibited, right? But the one that's not inhibited is going to be hexokinase 4, which is glucokinase. Okay. All right, and now 6. You guys got a bunch of D's. Great. Again, this is good. You guys got these right? Liver cells are showing a decreased concentration of glucose 6-phosphate. Okay. Right. And this is exactly what happens, to patients with, diabetes, right? Insulin promotes glucokinase production, right? So, when insulin's going out, the body's saying, okay, we need to store all this excess glucose and store it as glycogen. Okay. So if there's a lack of glucokinase, then there's going to be, total lower concentration of glucose 6-phosphate, right? And so this is exactly why diabetics have to be very careful with their sugar intake. All right. Does anybody have any questions about 6? Good job. Okay, let's go on to step 2 of glycolysis. All right, and this is called phosphoglucoisomerase. Okay, and so this is one of those super easy ones where it tells you exactly what enzyme class it is, right? This is an isomerase. Okay, and so this is, you can see over here that it's actually going to catalyze the movement of this carbonyl oxygen, this aldehyde, right? It's going to turn it into a ketone down here at C2, right? Okay. And so what this is going to do is actually going to set us up to being able to put a phosphate group onto C1 of fructose 6-phosphate in step 3. Okay. And so remember that question I asked about 2DG earlier, right? So 2DG removed this hydroxyl group from glucose 6-phosphate. And so, right, it made it so this 2DG actually doesn't even fit into the phosphoglucoisomerase enzymatic pocket. Won't even bind, right? So you can't convert this to F6P. Doesn't even bind. Okay. Okay, let's talk about step 3. All right, does anybody know what enzyme class this is? Almost. Ligase, you're thinking ATP. Good. It's a transferase. Right? And the name is phosphofructokinase. Right? And so kinase, right? It's what a kinase is, it's transferring a phosphate group from the the gamma phosphate group on ATP, right, onto a molecule. Okay, and I would actually, definitely know phosphofructokinase. This is a very important enzyme for regulation of metabolism. Okay. So step one, right? So this is step three. Step one was the step that committed the cell to taking up glucose, right? Puts the phosphate on, it's locked in the cell. All right. This step three, this is what commits the cell to actually metabolizing the glucose, right? Rather than converting it, to a different sugar or storing it, right? So I'm going to go back 2 steps, or maybe here, right? Glucose 6-phosphate, right, can actually be stored as glycogen. There's several steps that you can get glycogen, right? So once the glucose is in the cell, right, you can either, if there's not a need for energy, you can store it as glycogen for later, or you can keep going down glycolysis, right? And then step 3 can happen by phosphofructokinase, right? So this is PFK. It's abbreviated this sometimes, right? And so this step, it's committing it to metabolizing glucose rather than storing it. Okay, and this is an absolute massive major point of regulation in the glycolytic pathway. Step 3, and I'm going to even write this down, right? Step 3 is the valve. Right? That controls anabolic versus catabolic, right? Whether glucose is going to get oxidized into pyruvate or not. Okay. And this thing is regulated because it turns out that ATP itself is an allosteric regulator of PFK. Okay. And we'll take a quick pause. Someone wants me to go back to step 2. Got it? Sweet. Okay. So it turns out ATP is an allosteric regulator of phosphofructokinase. Okay, what does that mean? That means if you have, I guess we should draw it out, right? If you have phosphofructokinase, this enzyme, right? Here's the active site. ATP itself can actually bind somewhere else and alter the shape of this enzyme so it becomes less active. Okay, so what does that mean? That means when there's high energy in the cell, when the cells have high energy, high ATP, right? We don't need to go do glycolysis. We've got all the energy we need. We need to go store it. Equals lower glycolysis. Okay. Now, this is a super cool, this is very heavily controlled because if you have AMP, right, aka signaling that there's low energy in the cell, it actually acts as an allosteric regulator again, but doing the exact opposite effect, right? So it actually increases activity. Okay. So this would be like the classic allosteric regulation, curve. Right, you have the normal substrate to velocity. And then you have your allosteric regulators. Of phosphofructokinase. Got it? So PFK1. So this is a cool question. Someone just asked about PFK1 versus PFK2. PFK1 and PFK2, right? Those are symbolizing different isozymes. So back to this question, right? Remember when we were talking about hexokinase 1, hexokinase 2, hexokinase 3, hexokinase 4? What an isozyme is the same enzyme, the enzyme with the same function, but is expressed differently in the genome, and it might be more active or less active depending on the condition that it is, right? So you might have PFK1, that's like the general phosphofructose kinase, right? And then we have PFK2 that might be the phosphofructokinase dominantly in the liver. Does that answer your question? And I'll go back to this. AMP increases phosphofructokinase activity, right? So you can imagine, here's our enzyme over here. Here's the step 3 enzyme. Here's PFK, right? If you have AMP, it actually binds somewhere else on the enzyme, right? Which will make this very, very much more active. So you'll get more fructose 1,6-diphosphate. Okay. Exactly. So AMP signals low energy, so you want to make more glucose. No. AMP signals low energy, so you want to oxidize glucose, Zoe. I think that's what you meant to say, but you don't want to make more glucose. You want to burn glucose and turn it into ATP energy. Okay. Yeah. Right. AMP is low energy, so we need to make energy. We need to make more. And so far, we've gone through 2 committing steps, right? We've burned 2 ATP. We've burned 2 ATP so far. Step 1 and step 3, we both burned an ATP. Okay. So now the next steps, we have to get them back, and we have to get them back. We have to get more of them back to make this whole thing worth it. All right. AMP and ATP, those are like the classic examples, but there's actually high citrate, which comes from the TCA cycle, actually will lower PFK activity. It also combines, so you can actually have, citrate on here too. Right, this enzyme is very highly regulated. Okay, does anybody want this? Should I move to the next step? Does anybody need this more? Any questions? It's a very important step. So AMP signals for more energy, thus increasing PFK activity. ATP signals high energy, thus lowering PFK activity. Okay. No, because the cycle doesn't need to burn more. Okay. Right. So if we have a lot of ATP in the cell, that means that there's a lot of energy in the cell, right? We don't need any more energy. We want to store energy. Okay. And so if you want to store energy, you take glucose and you turn it into glycogen, right? You don't burn the glucose, you're turning it into a glucose chain. Glycogen is just like a big long glucose chain, that's stored in your liver. Okay. And so if there's low energy, then that your body's going to signal, we need to increase energy. We need to increase energy. So it's going to activate PFK, because PFK is the valve that commits glucose from going down to being completely oxidized via you know, you get those net ATPs that we're going to talk about, but it also, you know, will send it down through the TCA cycle and, you know, do your oxidative phosphorylation. Okay. All right, so steps 4 and 5, right? These are less important step, or every step's important, but these aren't heavily regulated, right? We have step 4, which is fructose is phosphate aldose, right? And this is actually a lyase. Okay, which again is the non-hydrolytic and non-oxidative cleavage of single bonds, right, to form a double bond. Okay. So this step right here, it takes a C6, right, and it turns it into 2 3 carbons. Oops, so like this, it takes 6 carbon molecule and turns it into 2 3 carbons. Okay, so 6 carbon molecule, right, splits it into glyceraldehyde 3 phosphate and DHAP. Okay. Now, step 5 is this weird step, and we don't want DHAP. What we want is more glyceraldehyde 3 phosphate. So it just takes DHAP, right? And it uses this enzyme called triose, right? 3 carbon sugar, phosphate, isomerase, which is this enzyme, right? And as the name says, it's an isomerase, okay? To move the phosphate group down to the C3 carbon of glyceraldehyde 3 phosphate. Okay. And so now what we get after steps 4 and 5 is we get 2 glyceraldehyde 3 phosphates, 2 GA3Ps. Okay. Okay. And so now we have 2 of these, right? 2x. Step 6 is going to be, oh, sorry. Cool. All right, so in step 6 of glycolysis, right, this enzyme is going to be called, what is it? It's glyceraldehyde 3 phosphate dehydrogenase. What a mouthful. Does anybody know what class of enzyme this is? Oxidoreductase. Okay, it's because we're transferring electrons from NAD to NADH. So anytime you ever see a reaction where there's NAD going to NADH or NADH going to NAD, it's going to be an oxidoreductase. All right, just quick rule of thumb, simple. Okay, now I know that one. All right. And so this is, you know, step 6, right? we're taking the oxidation of an aldehyde, and we're turning it into a carboxylic acid, which is actually, usually favorable. And because it's favorable, you can actually couple that and add this phosphate group onto this molecule, right? So you'll notice now that 1,3-diphosphoglycerate is this 3-carbon molecule that has now 2 phosphate groups on it. Okay, and so because this molecule has 2 phosphates on it, we're going to say that this is a high energy molecule, right? Phosphates next to each other, they don't want to be there. They're willing to leave, and you can capture some of that loss of energy to, and store it in form of ATP. All right. Let me ask you a question now. What element is under phosphorus on the periodic table? This element called arsenic. Okay. And it turns out arsenic can look very similar to a phosphate. Okay. So it turns out if you eat arsenic in enough quantity, this enzyme, glyceraldehyde 3-phosphate dehydrogenase, can actually pick up this arsenic group, and you can actually turn that into 1,3, instead of 1,3-diphosphoglycerate, it can turn it into, let me just draw it out, right? It can add the arsenic group. Instead of this, it can be in, the O. Okay. And so why that is significant is because later in glycolysis, right, you're not going to be able to recover ATP. You're not going to be able to have a net gain of ATP because you have arsenic instead of a phosphate group, right? So this is why arsenic poisoning is so bad, right? It's a futile cycle. You won't get your energy output back. You don't gain after your net energy investment of 2 ATPs, right? This is something that you'd say, this is a reaction. If it comes up on the MCAT, this would be a decoupling reaction, right? So arsenic here would decouple glycolysis and prevent a net storage of ATP. All right. Okay, step 7. So this is the break-even step, right? So this is phosphoglycerate kinase. Right. Again, we're transferring. Kinase is the transfer of a phosphate group, and so we transfer back onto ATP. So we've just broken even. So remember, we invested 2 ATPs in the first half of glycolysis, right? And remember, we have 2x of these. So this reaction actually happens twice. So we get our 2 ATPs back. So now we haven't lost any ATP. Okay. Wait, no, what was that? Oh, yes. It's a decrease in the net storage of ATP. So we can look at it now here. Imagine if this was arsenic. Right? Step 7, you wouldn't gain any ATPs on it, right? You couldn't do that. Arsenic would prevent that. Arsenic's not phosphate, so you wouldn't get ATP back. And so you would not break even. Okay. Yeah, exactly. And you know, MCAT loves absolutely loves it. We're gonna go over another one that they love for gosh, I'm blanking out. In the electron transport chain, phosphate synthase, right? You can decouple the electron transport chain, and that's another thing that pops up on the MCAT pretty often. So just know what decoupling is and be on the lookout for it. Is it going to prevent energy, the net positive energy formation? Okay, and what does that mean for the body? Okay, let's erase this real quick. Again, and this will make more sense. There's another reaction that actually bypasses phosphoglycerate kinase, right? So there's a reaction that can bypass step 7 in glycolysis, right? And it can actually take 1,3 diphosphoglycerate and it can turn it into 2,3 BPG, right? Which is essentially just taking this phosphate group here and moving it down to here. And why this is important is because 2,3 BPG, right? This is actually, what you're not going to learn until later, is going to stabilize the oxyform of hemoglobin. Okay. So this is actually the molecule that's responsible for, the hemoglobin cooperative, binding curves, right? And so this will be covered in the heart and lung lecture. But it's good to know where these molecules are coming from. It's coming directly out of glycolysis. I don't want to botch it. I forget if it's the TRR. I could just Google it, right? And it'll show you that. But yeah, that is something that you should know. Okay. And one thing I also want to point out is for step 7, this came up a little bit earlier. Notice how the arrows in both directions here, even though we generated an ATP. That's because 3-phosphoglycerate, this is a really high energy molecule, right? And this is also a high energy molecule. Right? They're both high energy molecules, so they have less of a problem going back and forth, right? Every time you do ATP to ADP, it's the delta G for both of these is, right around equilibrium. It's close to 0. Okay. Okay. So depending on the needs of the cell, right, you might favor this. It might favor 3-phosphoglycerate. Okay. Does anybody have any questions on that? Nice. Yeah, I should honestly know that, but I don't want to botch it. Okay, step 8 and 9. Right? Can you guess what this one is? What enzyme step 8 is? It's a isomerase. We're moving this functional group to a different point in the molecule. It's okay. And that's why you just practice these, right? But this is like literally like MCAT question. The MCAT has, this will be a figure right here. And then obviously they're going to ask you questions about what this means about glycolysis and blah, blah, blah. But you'll get an easy questions like what enzyme class is it? And then just, if you know it, you'll get a free point and it's great. And someone wants to go back to the last slide real quick. Okay, and now step 9, right? This is an lyase, right, which is performs a dehydration reaction to get to phosphoenol pyruvate. Oh, sorry, this is a isomerase, right? And that's because we're transferring a functional group to a different point on the molecule. This is the C3, this is C2, this is C1, right? We're going from a phosphate group on C3 to a phosphate group on C2, but we're not changing the composition of the molecule. Oh, and in step 7, it's a kinase. You remember what a kinase is? Transferase, right? We're transferring a phosphate group from here onto ATP. And sorry for all the scribble scrabble. No worries. And yeah, don't, you're not supposed to have this already memorized, right? Or already known. I'm just presenting it to you. You're going to see, okay, what's important and what's not important. And then you're going to go do practice problems. And then you're going to have to revisit it. You're going to go read the chapter. You're going to go read your notes. And then you're going to slowly, actually know this stuff. But, lectures are just so you know what's important and relevant and how it relates to things. And of course, how the MCAT's going to present it to you. Okay. All right. So step 9, right? It's just, performing a dehydration reaction. So this is actually an H2O comes out of this. And it's turning 2-phosphoglycerate into phosphoenol pyruvate, which is an extremely high energy, right? This is high energy. Oh, the pink one looks good, doesn't it? Pink marker. Nice. Okay, step 10. This is the last one. So if you guys, let's see if we can get this one. What enzyme class is this? Which is a transferase. So it's good to get them wrong now, right? Okay. And so you'll notice that yeah, exactly. And I mean, this is, the enzyme classes, it just takes honestly just memorizing and looking at a bunch of reactions and guessing and then being wrong and then doing it again. Okay. Right. And so this is the payout step. We'll go back to this pink one. Okay, so we've already recouped all of our investment. And remember, we have 2x of these. And so we can actually get a net gain of 2 ATPs. Comes from this step. Right, and this has a very large negative delta G. Okay. And so for any of these steps where you're generating ATP or using ATP, it's a pretty good sign that they're probably a site of regulation. Okay. So specific to this reaction, effectors that inhibit this are again ATP, right? Right? Inhibits pyruvate kinase, which is PK. Acetyl-CoA does. Inhibits. Alanine does. Inhibits. right? And it turns out that, yeah, alanine's an amino acid. Yep, got it. And that's because alanine is the amino acid counterpart of pyruvate, right? So alanine is actually synthesized from pyruvate. Okay. Leave that stuff up here, give myself some more room. Ugh, just erase it. Okay, so it turns out that liver pyruvate kinase, right, it can be covalently modified. Okay, so glucagon, which is the hormone that's opposite of insulin, can activate this cyclic AMP dependent protein kinase to phosphorylate pyruvate kinase. All right, and what this does is it makes pyruvate kinase much more strongly inhibited by ATP and alanine. Okay, so you can imagine drawing, right? Whenever you hear, technical language like that, it's good to kind of get a map or a picture, especially when you're drawing out your from your passages at first, right? And they start explaining things to you, right? And so glucagon will activate cyclic AMP cascade, right? Which activates a cyclic AMP dependent protein kinase. Right? And this protein kinase can phosphorylate pyruvate kinase, which makes pyruvate kinase more easily inhibited by ATP and alanine. So I know that that is a lot of information I just gave you, and I just drew this whole pathway out. This is how MCAT passages are structured. They're going to describe. So how would I classify? So glucagon is a hormone that's going to signal a cyclic AMP cascade that's going to activate a protein kinase that's going to phosphorylate the enzyme pyruvate kinase. And if pyruvate kinase is phosphorylated, it's going to be allosterically, it's going to be a negative effector of allosteric inhibition, which means that in the presence of ATP and alanine, pyruvate kinase is going to be less active, which is a mouthful. But that's how MCAT, that pretty much anything like this little pathway that I drew here, where you have something that turns on something, that turns on something, that turns on something, that inhibits something. That's how pretty much every single bio MCAT passage is going to be. And it's your job to be able to draw this little pathway and know what the heck is going on. So if I were to, give you a drug or something that blocks this, a drug that blocks this, a drug that blocks this, a drug that blocks this, how does that affect the pathway as a whole? And that's super high level strategy to understanding an MCAT biology passage. There's going to be some sort of pathway that affects something. Yeah. And that's why we're going to do this when we go over passages. Okay. And so at first, a lot of people, I did it, and people do, you need to get it real when you start doing your full length test, especially, like, you're going to need to get really good at drawing these little maps based on what the passage is saying, right? And that way, if you block glucagon, right? This doesn't happen. This doesn't happen. This doesn't happen. This doesn't happen. So this isn't inhibited. Okay. So protein kinase is a negative allosteric inhibitor of pyruvate kinase? Yes. Beautiful. And protein kinase, there's so many different protein kinases. Protein kinase is a catch-all for a protein that phosphorylates something. And depending on what enzyme gets phosphorylated, that enzyme is either going to be more activated or less activated. And that's like probably the most common thing on the MCAT. It's, pathways like this, and something gets phosphorylated and that increases or decreases, a function. Okay. All right, and you guys are all troopers for staying this long. This is cool. This is a double lecture. Okay, so now we finished glycolysis. Okay, and recap, we've gone from 1 glucose molecule to 2 pyruvate molecules. Okay, so we have net gained 2 ATPs, right, and 2 pyruvates. Okay, what's wrong? What's wrong with this? We've done glycolysis. We've gotten to our pyruvates. What's the problem? What's out of balance here? What's out of whack? Something is out of whack. And what that is? Yes, NADH is out of whack. And what specifically is out of whack? If you look at the oxidation states of this molecule and these molecules, you'll notice that glucose has an oxidation state of +0, and pyruvate has an oxidation state of +2. And so what that means is our molecules are out of whack. What we've done is we've generated too much, NADH, right? Our balance is off. And so depending on what your organism is, depending on, whether it's anaerobic or aerobic, pyruvate's going to have several different fates. Okay. So do you guys know how to calculate oxidation state, by the way? And do you want me to show you? Okay, cool. This is good. These are, again, free points on the MCAT. So let's just erase. So here's your little oxidation state. Right, so oxygen equals, oxygen equals -2, meaning like a double bond oxygen. An oxygen in like a hydroxyl group where there's a single bond is -1. A hydrogen is going to equal +1, and a carbon is going to equal 0. Now, let's be careful here. That's the oxidation state of those molecules. Now, when we're getting the oxidation state of one of these species, you're finding the net oxidation state, of all your carbons. Okay, so if you can imagine, right? Let's just look at C1 right here. So you have a carbon attached to an oxygen, attached to a hydrogen, attached to a carbon. Okay, so we look down here at our oxidation state. Hydrogen is +1, which means the carbon gets a -1. Oxygen is -2, which means the carbon gets a +2. Carbon to carbon is 0 and 0, right? So this specific carbon, the oxidation state is -1 + 2 + 0, which equals +1. Okay? So far, so good? So if you wanted to calculate the oxidation state of this entire molecule based on its carbons, you could literally go down the line. So we just did this one, and that equals +1, right? If you go to the OH, right, this means that it's going to be a +1, -1. This is going to be a -1, +1. And so those are just canceling each other out. +1, -1, -1, +1. And then down here, right, this is just a carbon attached to a hydrogen in OH. So this will be +1, -1, -1. And so if you add all those up, right, all these things cancel each other out. We don't need to worry about all that. Right? The net charge down here on C6 is equal to -1. Net charge on this one is equal to +1. So the total oxidation state is equal to 0. Do you guys see how that works? Oops. Do you guys see that? Okay, so you're talking about right here. Right here. So this one right here. Yeah. So the oxidation state of hydrogen is +1. So hydrogen is +1. And so what that means is the carbon gets a -1 from that. Right? So if you, let's just draw this out again. You have a carbon attached to a carbon, attached to an oxygen, attached to a hydrogen. If the hydrogen is +1, the carbon is -1. If the oxygen is -2, the carbon is +2. And this is 0 and this is 0. So you circle these three, and that equals a total of +1. You got that? Cool. Cool. All right. So we're not happy yet, right? Our pyruvates, which again, if we did this, are going to be +2. Okay, something needs to happen in order to keep homeostasis. So here's pyruvate, and there's 3 fates of pyruvate. Pyruvate can either go to lactate, it can go to ethanol, or it can go to acetyl-CoA. Acetyl-CoA is probably the middle metabolite of all of metabolism, and this will get fed directly into the TCA cycle, right? And there's an enzyme here, or a complex of enzymes that goes from pyruvate down to acetyl-CoA called the pyruvate dehydrogenase complex, PDC. Okay. And essentially what that does is it does exactly what it says. It does a pyruvate dehydrogenase complex, right? It'll turn NAD to more NADH, right? And it'll turn coenzyme A into acetyl-CoA, right? So essentially you'll get a CO2 blown off of here. And so you've taken this 3C carbon and you're turning it into this 2-carbon molecule, acetyl-CoA. Okay, and you give off a CO2, and you're still accumulating NADH. And just like you said earlier, we're starting to produce too much NADH, and we're going to cover this in the next lecture. These are going to end up going to the electron transport chain for oxidative phosphorylation. Now, if we're not going to be doing oxidative phosphorylation, right, what's going to happen? Glycolysis, we're starting to accumulate NADHs, okay? And so somehow, if we're going to keep going through glycolysis when we don't have any oxygen, right, when we're anaerobic, I think I spelled that wrong. Anaerobic, right, no oxygen, right? In the presence of no oxygen, we somehow need to gain back our NADs, right? We need to regenerate our NADs, and otherwise we're not going to be able to continue to oxidize glucose into pyruvate. Yeah, you need to know fermentation for the MCAT. Definitely. Okay, so this reaction will take that NADH that we produced in glycolysis, and it'll turn it back into an NAD plus the H plus. Okay, and you'll notice that a lactate has gone back to an oxidation state of 0. Now, this is what happens in humans. We do lactic acid. I guess that would be fermentation. In organisms like yeast, what they do is they do ethanol fermentation. And so they'll actually have an intermediate acetaldehyde, and they'll regenerate it this way. Notice that it blows off a CO2 here in the first step, taking this 3-carbon pyruvate into a 2-carbon acetaldehyde. And then the next step is where you can regenerate NAD plus. Okay. Okay. Excuse me. Does anybody have any question about the fates of pyruvate? Yeah. So what we just covered was a whole lot of information. Someone wants to go back to step 8 and 9. Got it. Does anybody have any questions about this? Cool, great. So that's it for this epic lecture. If you guys want to, I can do this question with you guys. Yeah, my brain is fried too, trust me. Yeah, let's do it. I'll stick around. Might as well get it to 6 p.m. on the dot for a 4-hour lecture. And this is how the MCAT is. MCAT 7.5 hour seating time. You know, you're going to be fried and have to answer questions like this. Imagine that. We would just be finishing our lunch break in the MCAT and have to do the whole thing all over again. How messed up is that? Oh, it's bad. It's brutal. It's an endurance game. You gotta work up to it. It might be a little shorter than 7.5 hours. They've actually shortened the breaks since COVID. Same amount of questions, same amount of testing time, but I think there's actually less breaks. Oh, it's 8 p.m. for you. Yeah, I forgot. It's 6 p.m. I'm about to go right after this. About to take a load off, grab a beer, go watch the Giants and Dodgers game 5. Yeah, it's good. I mean, you guys are troopers for hanging in there. I swear it's a good sign, you know. And again, this is all so much information. You know, you've seen it now, and you know, now every time you interact with it, you'll be able to pull out the information easier. Yes, Alexandra, and we'll get into that. And the best way to do that is to just do practice questions, enough of them, and you'll start to make these little maps. And so this question that we're going to do right here is incredibly hard, and this is like creme de creme type of MK. If you guys can both get these right, if you guys can get these right now, I'll be very impressed, and you're going to be very in. If you can answer these and understand them this early, it's a really good sign. uh, yeah. Yeah, Jonathan, that's funny. It's true. It's a grind. Take another minute just so people can have time to try to actually figure this out. Good exercise to do that. Yeah, no rush. Try to like draw this one out, figure it out. Okay, cool. Let's talk about it. All right, so clearly there's a lot going on in this question. We're talking about mutations. They even mentioned NMR. They're talking about anabolism, catabolism, uh, multiplying, proliferating cells, cancer cells, whatever. There's just a lot of stuff going on. There's a huge figure with all these mutations on it. You know, what does all this mean? How do you like approach this? What's the right answer? Okay, so you guys guessed what, B and B? You guys guessed a bunch of B's here, right? Does anybody else guess anything besides B? 8, we got a C for 8. 8B, 8D. Ooh, we got an 8D. Cool. Okay. So, you know, again, the more you know about glycolysis, the quicker you're going to be able to understand what's going on, right? And so this passage is actually describing this phenomenon called the Warburg effect, which is something that happens in cancerous cells, where cells that have cancer, for some reason, or I'll tell you why, they start burning glucose, right? And a lot of that energy is not coming out, right? Meaning that they're using a lot of glucose, but instead of going strictly for ATP production, right, it's going towards anabolic pathways, right? So it's going to go to the pentose phosphate pathway. It's going to go to all these intermediates that come out of glycolysis to start building up cellular structures. Because remember, cancer cells are dividing. So they not only need energy to do that, but they're going to need a lot of carbon from that glucose to build, you know, new structures and to multiply and divide. Okay, so this Warburg effect, which I do highly recommend reading into, very, you know, fascinating, is this phenomenon where cancer cells will just be burning through a lot of glucose and not necessarily using it for oxidative phosphorylation through the electron transport chain. Okay, and so that's exactly what this passage is talking about. Okay, and you'll see that this passage, you know, is talking about a specific step within glycolysis, particularly pyruvate kinase, which we've just gone over, right? That's going from PEP to pyruvate and it's generating ATP. And it's saying that there's these two different isoforms, right? So you have pyruvate kinase M1, which is, in fact, I'm just going to highlight this. These, I would say, are very important, right? In contrast to quiescent cells that are not multiplying, right? Cells that express the continuously active isoform of pyruvate kinase 1, proliferating cells selectively express the M2 embryonic isoform of pyruvate kinase, PKM2, which is regulated by phosphorylated tyrosine phosphorylated proteins. Okay, so just as you're going to learn this actually in cars, and it's weird. There's this weird overlap between cars and biology, and there's certain things you got to look out for. Like, you ever see like a in contrast word? That is probably a pretty decent idea that they're going to be comparing two things that are important. Okay, in this case, it's comparing PKM2 to PKM1, right? And it's saying that, did someone say something? And it's saying that PKM2, maybe I should write this on a blank page, right? It's saying that PKM2, embryonic pyruvate kinase, right, is regulated by protein kinases. Whereas PKM1, which is the normal pyruvate kinase, is always on. Are you guys with me so far? Awesome. So this is really good, right? We've looked at it. We know there's these two different ones. One is always on and one is getting turned on and turned off somehow by this protein pyruvate kinase. Okay. So, oops, let's go back up here. So now what are we interested in, right? What are they doing? What are they testing down here, right? So when, activated by this growth factor, FGF, right, this growth factor receptor is known to phosphorylate PKM2 at tyrosine residues. So now we know that these protein kinase is phosphorylating at these different tyrosine residues. And then it lists a bunch of different residues that it could get phosphorylated at, okay? And so what they did is then they did this mutation analysis, right? And then they wanted to record the relative activity of PKM2, right? If they mutated each of these cell lines at different tyrosine residues, okay? So they turned tyrosine into phenylalanine, right? In different cell lines. And then they measured the relative catalytic activity of PKM2. And so you'll notice, right, that for all of these residues, there's only one of them that showed a significant difference, right? And that's saying if I changed tyrosine to phenylalanine in this cell line, right, at residue 105, right, in the presence of PKM2, in the presence of FGF, right, I'm actually going to get an increased catalytic activity. So the enzyme is turned on. Are you guys with me? Okay. And everything else, nothing happened. It's not lower, it's not greater. They're all within the error bars of the wild type, which is the control. So these ones don't matter. Okay. So, maybe we should draw this out, right? Again, we're on the MCAT. So it's good to, you're going to have to come up with these techniques, how you tackle the bio passages. So let's draw out what we just said, right? We've got this fibroblast growth factor. So FGF which is some sort of molecule or peptide hormone or something, right? And it's going to come into contact, right? Here's a cell membrane, right? With this receptor protein. This is called FGFR1. And if this thing comes into contact with the receptor protein, then this receptor protein then activates or phosphorylates protein kinase, right? So we'll get this phosphorylation of this protein kinase protein, right? Which just happens to be pyruvate kinase in this case. Okay. And so what we just did, right, is we noticed that going back down one, oops, up one, sorry, that if we got rid of this residue here, right, so if you get phosphorylated at this tyrosine residue, and now all of a sudden you're a mutation where you don't have this tyrosine residue, right, this is not going to happen. You're not going to be able to phosphorylate. Oops. Maybe I'll draw this a little clearer, right? So you have your protein, and we'll draw it green. And you have this tyrosine residue, right? Y, that can get phosphorylated, right? So if you mutate this enzyme, and so it does not have this tyrosine, right? It's replaced by an F. You're not going to have it phosphorylated. So you're going to have, oops. This enzyme. And so now you have to ask yourself, is this enzyme active when it's phosphorylated or not active, right? And what's the answer? It's actually the opposite. It's deactivated, right? So this equals less active. And this equals more active. Okay. And so you can imagine, right? Again, we're talking about PKM2, right? And the difference between PKM2 and PKM1 is that PKM1 is always on. And that's because it doesn't get phosphorylated here at this residue, right? So this one's always on. So this one's always converting PEP into pyruvate, right? Which goes into oxidative phosphorylation, right? And so the significance is that PKM2, right, it's a different isoform of PK that is able to be regulated by phosphorylation, right? And so the idea is that in cancerous cells, which are proliferating, right, they want to be able to control glycolysis, and they want to be able to hijack glycolysis and say, okay, we don't only need energy, we need to start doing anabolism. We need to start building up our, nucleotides. We need to save carbon for all the structures we need to build, as well as energy, okay? And so that's why you'll notice in cancerous cells, you'll get all of a sudden this isoform popping up, right? PKM2. All right. Does anybody have any questions about this? This is very tricky stuff, right? There's a lot going on. You have to know glycolysis. You have to know, receptor proteins. You have to know phosphorylation proteins or kinases, right? Lots of stuff going on. Does anybody have any questions? So let's go through these answer choices, right? What is this saying? Which cell lines expressing PKM2 would you expect to find an increase in cell proliferation in the presence of fibroblast growth factor. Okay. So which ones do you think are going to have an increased cell proliferation? The answer is these ones. Right? Because this one turned it on more, right? If we mutated it to keep it always on, like we see here in Y in the tyrosine 105, the only one that's significant, by the way, right? We're turning that on all the way. So we're going from PEP to pyruvate down into oxidative phosphorylation. Okay. Now, if we have just the wild type one, right, where it can still be regulated, you're actually going to be in the wild type, right? It can still be phosphorylated, meaning it can be turned off, meaning you can go through those anabolic pathways. Do you guys see that? So this is why this is such a tricky question, right? Because it makes you, this question is designed to make you think this one, right? And so the idea here, yes, but what it means, right? So no, yes, right? Psych and Soc, you're going to get significant value graphs all the time. For biology, you are too, in both. In fact, all sections, the MCAT besides CARS, right? And so, you know, the significant, this means that you can compare, oops, let me go to a different color. Let me erase this. And when you see a significant value, right, it means that this is going to be different from everything else, right? So that's definitely the place that you want to look first. You want to know what that means, right? Why is this significant? And in this case, it means that if I get rid of this amino acid that can get phosphorylated, I'm going to increase my enzyme activity, meaning this mutation right here, Y105 to F, is essentially making PKM2, it's turning it into PKM1, right? Are you guys getting it so far? Yeah, exactly. And that's why this one can be bucketed in for question C. It's because these two species, there's no significant difference between them, right? So you can treat them essentially like the wild type, right? And in fact, even though there's no significance, it even shows less activity. So this actually might help regulate even more, right? But you don't have, the data doesn't say definitively. Does that make sense? Yeah, you could look at it that way, exactly. Right? And so again, going back to every single passage, I want your eyes to go straight to wild type. I want you to go, okay, first thing you look at is control, wild type. Second thing you look at is what's changed. And then third thing you look at is what does that change mean? Okay? Does anybody have any questions about the answer for this question? Again, this is an incredibly hard, very realistic MCAT style question that I don't expect any of you guys to be able to do these right now, but this is where we're moving towards. This is the end game, that you get these right. And then you can get it right quick enough, right? Because you realize you're going to have not much time for this. Cool. So good question. Exactly what are we looking for with PKM2? So for PKM2, right, we're comparing PKM2 to PKM1. PKM2 is an isoform that is expressed more in proliferating cells, right? You can think proliferating cells, you should have a little ding, ding, ding go off and think, cancerous cells. Cancerous cells are proliferating, right? And so the whole idea behind this Warburg effect is that you need to keep that carbon and use it to build up molecules, right? And some of the molecules that it needs are things that come out of glycolysis directly, so like amino acids, 5-membered rings which go from glycolysis to pentose phosphate pathway. You're going to need NADPH, right, which we're going to talk about later, so don't freak out yet. But this is actually the electron carrier that is used in anabolism, right? So when we're talking about fatty acid synthesis, NADPH is actually what gets oxidized to reduce fatty acid chains. All right. Does that answer your question a lot? Kind of, yeah, I know. Right, let's go back to this. Oops. This is what you're looking for, right? You need to understand that if PKM2 is phosphorylated, it can be regulated, so you can actually turn it down. You can make it so it doesn't turn PEP into pyruvate, right? And so if you take away its ability to be phosphorylated, now all of a sudden it's continuously on, right? Which is similar to PKM1. Cool. Yeah, exactly. And so these single amino acid swaps that you saw here, these are going to be very common on the MCAT. And so that's why I said in the last lecture, that you need to know your amino acids, right? And it turns out that tyrosine, God, I'm blanking out on the structure, right? But you have you have your residue, and at the very bottom of tyrosine, I think it's a ring, and it's got a OH group on it. Turns out tyrosine, tyrosine is very common for getting phosphorylated. So it's actually an amino acid that if you do single amino acid swaps, it messes up these entire cellular pathways. Okay, cool. Let's go to question 8. We haven't even gotten to question 8 yet. My God. Let's talk about this. Does anybody have a guess for this one now? Now that we've understood the question, let's try to answer 8 now. Oh. Okay, so we've got got it all over the place. So the answer is actually D. Yeah. Okay, and why is it D? And why is this a really good question? So this is a good question because, and this is why you need to know your glycolysis, this is why you need to know your metabolic pathways, is because it's asking, cellular concentrations of, metabolic intermediates, right? Lactate, right? That's the end of That's lactic acid fermentation, right? NADPH or NADP, sorry. This is the electron carrier that turns into NADPH in the pentose phosphate pathway. And, you'll learn about that later, but, for now, right, you can eliminate it because it says we need a lot of NADPH. Okay. Right here, we're asking about concentrations of phosphoenol pyruvate. Okay. So why is the answer D? Okay. Notice how in the question, now we're talking about PKM1 only, right? Right. So we're talking about the FDA just approved a drug, okay, that is going to cause some sort of cellular dysfunction, right? And are they going to want to cause a cellular dysfunction in normal cells? Are they going to want to stop glycolysis in normal cells? No, of course not. And so you want to design your drug to work on only cancerous cells. Okay, so the drug acts as an FGFR inhibitor, right? Oops. Down here, right? So it prevents this from binding to the FGFR, right? So it essentially stops the phosphorylation of pyruvate kinase M2, which means that pyruvase kinase M2 will be more active, right? And so if you're a cancer cell, you don't want to be more active. You want to be doing anabolism. You want to be building up your cells so you can, divide and proliferate. Okay. Does that make sense? And so that's why the answer is D. Anybody? Yeah, this is it's too much, honestly. This has got to be 2. Okay, why not C? Good question. Increase in cellular phosphoenol pyruvate. So let's go back up to, here's pyruvate kinase right here, right? Would we get an increase of this? Right, so we're talking about PKM1 right now, which is always on. Remember, this is always on. So we're taking PEP and we're turning it into pyruvate all the time. So I don't think this drug is going to stop that. So I think that this very unstable high energy molecule is still going to be getting turned into pyruvate. Can you vibe with that? Cool. Now, the only one that I would think would be a tricky question, and the one that would trick me is this one, right? Increase in cellular lactate, right? Need to get into it too much, right? But wait, if PEP is being turned into pyruvate, right? Isn't a fate of pyruvate lactate, right? Wouldn't that increase lactate concentration? That's what, trick question. But the cancer cell still needs to get energy, right? So it still needs to coop up energy from this as well. And it prefers to go to lactate because lactate doesn't burn off any carbon. Whenever you go down to, and sorry for this total mess in here, right? Normal cells, right? When you go into acetyl-CoA, you burn off a CO2, right? In exchange for, down the road, you're going to get, increase your ATP. But when you burn off CO2, right, that's very much so catabolism, right? You're completely oxidizing these carbons in exchange for energy. Lactate, you get some energy out, but you still have your carbons, which means this thing can go backwards and go down all these other pathways that will build up cellular structures. Do you guys vibe with that? Cool. Yeah, and again, you guys are going to just need to decompress, and then, we'll revisit all this stuff. That's just the name of the game. Hearing it again, doing practice questions, doing more practice questions, and then you'll soon get really quick at this. But I hope today you guys were able to get a little bit of a snapshot of why all this stuff is important, right? And what all these things mean, and how everything, and things are regulated and this and that and blah, blah, blah. Cool. Well, I think that's it. Does anybody have any questions? Want me to go over anything? Yeah, I think we all need a break. Brain go burr. Yeah. Yeah, I hope it's, yeah. Yeah, I'm sorry. This is it's a big old lecture. Yeah. And we can definitely do that, and we can work on it in office hours. And questions are going to be your number one friend now, right? Because you're going to hit questions. You're going to be like, oh, we kind of went over that lecture, did we? What was that? And then, then you're going to relearn it again and understand it this time. Yeah, of course. That's a really good question. So phase ones are supposed to feel slow and painful, and they take forever, and you don't do that well on them. The class averages, if you can get 50% right on, do you guys see that? It's on the portal somewhere. There's like a schematic, right? Here, there's like a graph somewhere, and it's like just a schematic, but it's pretty accurate actually. Yeah, of course. Where you get like percent of questions correct. Or no, that's MCAT score. How does it go? It's a percent Q's correct. Oh, no, I got it. This is me being fried. It's both, right? It's MCAT score, TBR Q's correct. And then you have like the phases. 1, 2, 3 FLs, and that's it, right? And you're going to see this like huge increase in this second one, kind of plateau here, or sorry. The phase one is going to feel very hard, and then you're going to get this big increase in phase two, right? Into phase two, then phase three, you're going to plateau, and then full length, you're going to get this huge increase, and your MCAT score is going to be around here, right? And so phase questions, I think it's something like 40% is normal, 50% is actually really good for phase one, right? 40 is normal, 50 is good, and you're going to want to slowly get this up to around, 50 to 60, and then by phase three, hopefully around 70, right? And then FLs, this is its own beast, and we'll talk about that later. In between, you're not going to have time to reinforce content in between phase 1 and 2, right? I mean, they're like supposed to be like a week from each other, and you're going to have so many different other lectures to do. And the whole idea is that you're not reinforcing, you're like forgetting. Phase 2 is the reinforcement, right? Yeah. Like you're supposed to forget about it, right? Because, you know, if you study just one subject, of course you're going to be good at that subject. It's all about studying it, forgetting about it, relearning it, or trying to get them right, you know, next time you see it. Yeah, exactly, Alexandra. 50 to 60 is money. If you can get up to 70% of TBR questions, like on a big sample size, you're going to be really well off. Really well off. Of course. Yeah. Thanks for sticking around for so long. You guys are troopers. Cool. Sounds good.